Okay. Egunon, buenos días, good morning, guten Morgen, bonjour, everything. So many languages here represented. Ongi etorri, welcome to everyone, and a special warm welcome to those that are coming from abroad to San Sebastian. I hope you enjoy the city, especially with this excellent weather, which is not so common. If you know the place a little bit, it rains a lot, so very good luck today. So LUCIA is a project of the Cancer Mission in understanding lung cancer, the risk factors and their impact for better prevention, screening, diagnosis, and also to guide future treatments. It is a multidisciplinary effort of clinicians, biologists, engineers, social scientists, lawyers. So we need to speak together to understand what's going on, to understand what kind of solutions we can develop. So when we were at the kickoff meeting, I remember in a dinner with Unai, Dr. Unai Jimenez from Cruces Hospital, he suggested, okay, why don't we do some workshops so you understand what are the clinical challenges for LUCIA, so we settled some common ground for everyone. So we thought it was a good idea, and because we wanted to complicate it a little bit more, we thought that the same on the contrary. Why we don't show you how we engineers work, what we can do, and then we try to speak a common language for this project. Okay. So and of course opening it to the public in this workshop. So first, I would like to thank the efforts of the different speakers in preparing the presentations, the organizing committee of Bicontech and Biocruces, Bio-Vizcaya, the coordinator team at Technion, Hope for helping in dissemination, and very especially our communication team at Bicontech, which had a hard work in preparing this, which I know is very difficult. So after this opening by myself and Eunate Arana from Biocruces, which she will speak next, we will have a session. First, we will present Professor Hossam Haik by Technion Institute of Technology, the coordinator at Haifa, followed by two sessions. One will be focused, as I said, on the clinical aspects of LUCIA, and the other one more on the technological aspects on LUCIA, okay, with a break in the middle. Now, on behalf of Biocruces, Bio-Vizcaya, I would like to present Dr. Eunate Arana, who will make an introductory talk of the relevance of LUCIA project clinical objectives. Eunate Arana is currently the coordinator of research scientific proposals in Biocruces, her research institute, now Bio-Vizcaya. She's a specialist in epidemiology and primary care. She has clinical experience in both primary and emergency care and over 20 years of experience in evaluative research, particularly in the fields of clinical epidemiology and health outcomes. She's leading in Osakidetza 4 Horizon Europe projects related with public health and prevention of non-communicable diseases. She's also responsible for coordinating the research in colorectal cancer, colorectal cancer screening program of the Basque Country. She currently coordinates several research projects funded in competitive calls and other projects to improve the adequacy of clinical partners in the Spanish health system. So she's the right person to tell us the main objectives or the main goals, clinical goals of LUCIA. Thank you.
Thank you, Ivan, and welcome everybody. Yes, please. Okay. I think that all of we are here, we know which is the burden of disease related to lung cancer, and we know the impact, the social impact, health impact, and economic impact of lung cancer nowadays. But the problem is that lung cancer is growing up every year, everywhere in the world. We know that it's the second leading cancer for men and the third for women. We know already that the deaths related with lung cancer are more than the sum of breast cancer and colorectal cancer deaths all over the world. And we know that the figures for 2030, they are really terrible. It's going to be increased by 38%. the prevalence of deaths related to lung cancer. Yes, please. Okay. We know already that there are a lot of, and we know that which are the risk factor, the main risk factor related to lung cancer, and that most of them are preventable already. So I think the main gaps in order to find in the cancer, lung cancer, and most of cancers, and that it has to be a lot of efforts in the quick of smoking intake. Also, advances in new treatments, and one of the main gaps is nowadays the screening methods in order just to do an early detection of lung cancer. It's already known, not only for lung cancer, but also for other cancers, that early detection is crucial in order to improve survival. But in lung cancer, only 15% of patients are diagnosed at an earlier stage. They are being running out a lot of research studies all over the last decades in order to identify which are the best screening tools for lung cancer screening programs. In 2011, it was published this clinical trial that assessed that low-dose CT are a better screening tool compared with CT radiograph. There are a lot of guidelines nowadays published all over the world related to the implementation of lung cancer screening programs, and also the European Commission has addressed that it has to be implemented in European member states. We know that it's cost-effective, the screening program with low-dose CT scan, what is combined with smoking cessation. But nowadays, we have some gaps. One is that it's only related with two variables, mainly with two variables, the screening programs, with age and with smoking intake. But there are others risk factors that should be identified in order to do better programs, more efficient programs. And we know also that it's not only smoking the main gap related to the development of lung cancer, because nowadays 50% of new lung cancers are related and diagnosed in smoker or never smoker people. And another gaps are the false positives and the harms that the screening programs make nowadays. In this publication, we can see that in the first one, is made in Canadian screening program, the 96% of the positives that they were addressed in the CT scans at the first round were false positives. And in Nelson trial, this publication is from 2017, 75% of the participants have at least one incidental finding. In those, only 8% were clinically relevant, and 7% of these goes to invasive procedures that they were not needed, sorry. And another two main gaps is that CT is a harm identification tool because it makes radiation to people that probably they don't need this kind of tool. And another main gap is the cost of one CT scan for some health programs, health systems that maybe they can afford a CT scan as a screening tool. So in these gaps is going where is going to work the LUCIA project, trying to improve these gaps that nowadays are running out with lung cancer early detection in Europe and in all the world. So thank you very much.
So next, I would like to introduce Professor Hossam Haik, who is project coordinator of LUCIA. Professor Hossam Haik is full professor at the Technion, Israel Institute of Technology, and Dean of Undergraduate Studies. He's an expert in the field of nanotechnology and non-invasive disease diagnosis. His studies include the research and development of nanoarray devices for screening, diagnosis, and monitoring of diseases, nanomaterial-based chemical sensors, electronic skin, breath analysis, volatile biomarkers, and cell-to-cell communications. He has, in record, more than 400 publications And the technologies developed by Professor Haik and his team have led to the production of more than 52 patients. Some of these have been licensed to several companies and are ground for startup companies. He has received more than 82 prizes and recognitions, and he was also included in more than 42 important ranking lists, such as the one of the world's 35 leading young scientists by MIT and top 100 innovators in the world. So, thank you, Professor Haik.
Thank you very much, Ivan, and thank you very much for the Vicomtech team for the wonderful arrangement and the coordination of this day and the next 2 days to come. So we truly appreciate your efforts, dedication, and also the quality of the arrangement. And also, this is a good opportunity to say thanks for the technical team, to Dr. Yoav Broza, who really runs the project, the executive person in charge of this project, and Liat Tsuri, who are running things very nicely from our side during these times where everybody is busy. I would like also to thank all the partners of the LUCIA project, which will introduce within a minute for the dedication and the fruitful discussions in order really to try to contribute to understanding the lung cancer risk factors and their impact on the health, society, and citizen in Europe. And as we have heard from Onate from the brief introduction and really the one that summarized the major challenges in the field of lung cancer, the lung cancer is one of the biggest and deadliest cancers in the world. And if I take the numbers which Onate has mentioned, 3 million deaths per year, and try to divide per seconds, that means that we have one person that dies every 30 seconds in the world from lung cancer. And this is really high frequency, and these are really very, very big numbers. And furthermore, I want to iterate about one of the findings that has been mentioned by Onate, that the 5 years survival rate, namely the survival rate during the 5 years after the diagnosis of cancer, ranges between 15% to 21%. It depends on which year you read the statistics. And this also varies between men and women. For men, they die more than women from the disease. It's 17% for men and 24% for women. And furthermore, the lung cancer also depends on which kind of lung cancer the person has, whether it's small cell lung cancer or non-small cell lung cancer. And in between these subcategories, we have also a lot of variances in terms of the survival rate. And the question is why we do have such low numbers in the survival rate. At the time, if we take other cancer types, such as the breast cancer, which at the time had very low survival rates, today they do have some management ways and treatments to increase the numbers of the survival up to 90% or more. And I would like to say that the reasons for these very low numbers might stem from several factors. The first factor is called the infrastructure and the technology that is currently available for the use of the clinicians and the patients. This is in terms of the technology, infrastructure, and management issues. And the other critical point, it's more fundamental, which is the risk factors, which is the topic of our project. But beforehand, please let me just describe what are the real challenges in very simple words. And please allow me to see these words from the perspective of an engineer. I'm not a clinical doctor, so the clinical doctor can elaborate more about this point. One of the major issues with the lung cancer, as many other types of cancers and disease, is the misdiagnosis. And the statistics shows that the field might miss between 15 to 30% of the cases of lung cancer, mainly when they are still at the early stages of the disease. And if I would like to simplify things more, And to say it in really very simple words, sometimes you might feel healthy, but that doesn't mean that you don't have early stage of the lung cancer. And at that stage, there is a huge difficulty to detect the cancer. And that's really one of the reasons. And the other reason is the accuracy, specificity, and sensitivity of the currently available techniques, which we have in the clinics used every day. We have heard from the introduction of Funate that the CT is a great machine. It's a great tool to detect cancer, but it really has a lot of also false positive and false negative. For example, the CT can give an image of the cancer itself or the tumor itself, which is great, but it cannot know whether it's malignant or benign tumors, whether we want to intervene at time or not to intervene at time. And therefore, always we have more efforts that has to be done in order to increase the survival rates. The other issue is the interface and the collaboration between the different parts of the healthcare system. If I take a satellite and take a look on the healthcare system from above, I would divide it in terms of the lifetime of the patient to 4 main stations. The first station is the healthcare system, which everybody goes there, and it's the local clinic, which the person starts his or her journey from that site. They come to the clinic, they have some complaints about their health, and from there might be a suspicion about their health issues or maybe cancer, and then they have to be forwarded to the healthcare professionals. And during this timeline, of course, we have a lot of costs, we have a lot of waiting. I would just summarize, we have a lot of bureaucracy. Because of this bureaucracy, also the journey of the person during this circle of travel from one station to the other station is really long and cost, and till getting to the bed in order to get the treatment, this might consume a lot of time and a lot of effort, which might be really very crucial in order to give treatment at time and might save the lives. So this is what we call the management. And I would say that the border existence between the different parts is really very challenging. And I would like to imagine a healthcare system without all of these borders, where the interface between all of them are more efficient throughout AI, throughout data management, and also by the use of more efficient tools and diagnostic tools as we will use in the LUCIA project. And what I have said in the early slide is really supported by numbers. If you take a look on the numbers, what is the role and what is the contribution of each of these failures that might lead to the bureaucracy and to the delayed diagnosis of lung cancer, you can see that 63% of the patients have at least once experienced problems with assessment issues, which you can read what are these assessment issues. And you can see that 23% has some patient factors that have affected their getting the diagnosis at time. And also, please have a look about the 20%. 20% we have a communication problems between the different healthcare professionals and between the patient and the healthcare system and so on and so forth. Namely, only by 20% we can really do a lot by making better communication. And of course, insufficient documentation or lack of documentation, this is something that is really considered to be a big issue and bring a lot of hurdles in the early diagnosis. And all of that can be summarized into these numbers of how much the healthcare system lose every year in the context of the lung cancer and delayed diagnosis of lung cancer. Though what I have presented here are real challenges that everybody feels in the European Commission and the other countries, we have to think about what really can be the measure and the root of the problem. What is the root of the problem? If you ask me as an engineer, I'm not an expert in the cancer, I would say that we don't understand what is really the origin of the cancer per se. This is one of the major issues. We might have hints, we have hypotheses, we have theories, but really the point where the cancer will start its journey in the human body, it's really very challenging to understand right now. And much more, we don't understand totally what are the risk factors for the lung cancer and those that might contribute to the lung cancer. And I will give you one hint. For many years, we have thought that smoking might be the biggest risk factor for lung cancer, which is absolutely correct. But how you explain that 15% of the lung cancerous people are not smokers? So this is a different issue. There is something hidden beyond the risk factors that we see and experience every day. And therefore, we have in LUCIA put our aim to establish a wider perspective and a wider look over the risk factors and the subcategories that might lead for the lung cancer. And this will be great contribution to the scientific committee, because if we know the lung cancer risk factors, we can at least as a society, as a citizen, and as policymakers, we can at least reduce those risk factors or try to reduce these risk factors, so we will enable either prevention actions or at least to highlight those which we might detect at early stages of the disease, where, if we do so, the survival rate can jump from 20% to 70% only by early detection. And remember one of the messages to take home. Early detection is one of the best treatment ways today in the current area. You can improve it from 20% to 70% only by early detection. And this is really huge numbers. And for that, I had the privilege, of course, to partnership with 21 groups and researchers from all over Europe, from 9 countries, to establish this LUCIA project that aims to understand the risk factors that affect the lung cancer and their subcategories. And as you can see, we have in this group 6 clinical partners, 6 academic institutes, 6 SMEs, 1 industry, 1 research center, and 1 research and development center. All of these came together to give different angles of the solution that we are seeking to detect the lung cancer risk factors. The project is really complicated. It has a lot of layers horizontally, and it has a lot of perspectives also vertically. But I would like just to bring all of the project into one slide, which you see it here, that what we are aiming is to solve the management issues and the infrastructure issues and the lack of accessibility in the daily life, and to bring it to the human body. Let's imagine that we will bring some arms from the clinics into the human body in their daily life, into the wide population. And we will do so by utilizing very small and very tiny and wearable devices, which you see here, and you will hear more about it from Nanos Medical today and from the Technion team, that we will bring multi-omics, breath analysis, and this spectrometry into the field, into the population, in their homes or in their clinics before they come to professionals, to try to identify, together with AI models that will be built based on retrospective data, to try to build a model to see whether that person has a risk to get a cancer or any chances to get a cancer or lung cancer. And in this group, we will include both smokers and non-smokers because, again, smoking, it seems, not only the risk factor for lung cancer. And at this stage, we will do the general population lung screening. If we identify people that might have the chances to get lung cancer, we then forward those specific people to next station. We will do more comprehensive analysis, which will include also the same tools which I have mentioned, which can work also inside the clinics, but this time we will include the low-dose CT or CT without the low dose. Depends on what is the availability in the clinics. At this stage, we will do what is called precision screening. And once we will get positive screening and higher risk and with more validity of the result, we will go to the diagnosis stage, where we will use the same data together with a retrospective analysis in order to make really the early detection and the early stage of the disease identification. At this stage, we will not only make only diagnosis, but rather we will do a little bit more. We will include what are the risk factors that has been discovered during this journey and that has led for the detection of the early diagnosis. And during this whole pathway, you can see that in all the steps, we will assess the risk factors, and we will try not only to assess risk factors, we will also try to understand the cellular processes throughout part of our colleagues and partners. In this case, whatever we validate, namely the risk factors which we will validate at this stage, will be examined in animal models and in vitro in order to understand the cellular mechanism and the genetic issues that relates to this lung cancer. To be more specific about the objectives and the ambitions of the LUCIA project in more scientific terminologies, here we want to detect three angles of the problem of the risk factors. The first one is the personal risk factors, the other one is the external risk factors, and the other one is the cellular processes that relates to the human body or to the disease per se. So the specific objective number one, where we have started on January this year in our kickoff meeting, is to identify databases, namely some records, some historical data of people that has lung cancer and equivalents with control groups in order to see how and who has got the cancer. And these databases will include a lot of information about demographics, lifestyle, and exposure. And together with this, we will build a toolbox that says if you have this combination of factors, theoretically, you might have an increased risk for cancer. So just imagine that we build a theoretical model that says who has the risk factors, and then we apply this theoretical model to the population to identify theoretically, without right now any kind of hardware, who might have the risk for cancer. Then we might take all of these risk factors and validate those with the hardware and the new techniques which I have mentioned earlier. So this is our first step, and it's very important to identify these databases, and I'm really very glad that we have really very nice databases from different partners. And the next stage after this period is to talk to healthcare providers and the policymakers to see what their expectations, namely how they will use, how will they manage those risk factors, and what are the priorities of the risk factors identification, so we can just give them practicality. Please allow me to emphasize one point. Though our aim in the project is to understand, please just remember that we are also in a consortium that we want really to implement also our findings in reality. And therefore, the implementation is a very important part of this whole project. And therefore, understanding and implementing the understanding issues to the project requires us to advise with the patients, citizens, policymakers, and so on and so forth. At the later stages, at the second stage and the first stage, which I have mentioned, we have to use and develop further some portable devices and wearable devices that can bring arms of the clinics into the daily life of the patients or the people that might be at risk for disease. And for that, as I have mentioned, we will use several technologies that are really innovative ones. The first is the breath analyzer, which everybody can do even in the street or even in the supermarket, even though we will not do it in the supermarkets. But the person can exhale for 1 to 3 seconds into the device, and the device will be able to tell whether the person has cancer or chances or probabilities for cancer. More about this device you will hear later today from Usnat Salatavor, who presents Nano's company. The other approach is to utilize wearable devices which you can stick into your hand or to your chest, and it can detect a wide variety of biomarkers that are exerted from the disease or as a result of the disease into the skin or into the sweat. And from there, we can detect those biomarkers, and we can align and connect those with the cancer. And the other device, which we will utilize it later on for advanced stages, is a kind of mass spectrometer, which can give you a sequencing of the biomarkers that are related to the lung cancer by really simple, portable, and small manner, as you can see here on the screen. After we have developed these devices and implemented these devices, and we have started to get data retrospectively, namely the historical records, and also the prospective data which we have got from these devices in the clinical studies, we will try to combine the data together to try to get a model that might give higher accuracies for the diagnosis and detection of the disease itself. And later on, the other objective which we will have in mind is to understand. And this is really critical. After we define, again, the risk factors, we have to validate that these risk factors are real. And the best way to do it is in vivo and in animal models. And also to try to understand what is the source, namely how they affect the cell from the beginning and how they advance till cancer. And this is really very complicated work, but we have real experts and outstanding experts that can help us to do so in this endeavor. And of course, the major aspect of this project is to validate the risk factors and also to effectively disseminate the project results into reality in a way that can really provide synergies with EU-funded initiatives. And indeed, in this project, we will not work in a vacuum as LUCIA. Within 2 days, we will collaborate and we will meet here with the rest of the Cancer Consortium in the European Commission in a project that we have coordinated here as well, in order really to leverage the collaboration with other projects on cancer. So we will have a kind of mega cluster of discussions so we can gain more comprehensive understanding even beyond the LUCIA, and this is something on our agenda. The project has 7 work packages. 2 work packages relates to the management and the dissemination and communication. And here you can see that the scientific and technological ones relates to the building AI solutions for around the human in order to build those models, which I have mentioned earlier. And this will continue with work package 2, which have interaction with work package number 1, to build the software ecosystem that provide analytics and the applications that can be accessed by the clinical doctors in their daily life. And this is a kind of toolbox that everybody can use later on. So they can say, if I have this combination of risk factors, okay, does I have chances to get cancer in the future for a given person, according to some data that were collected on that patients. And again, you will hear more about this ecosystem later on and about the vision behind it later on. And as I have mentioned, the work package number 3 will talk about the technologies that will access the healthcare system and the management ways into the population, and work package number 5, which connect with all of the rest of the work packages, is to identify the molecular risk factors, namely to understand the risk factors at the molecular level, which is quite important and critical for this project. And the validation by clinical studies is quite important and will be done in work package number 4. I will not go into this flow chart, how we will do the analysis and how we will understand the risk factors, but you can see that it's a combination of all the work packages together. It's an iterative manner between the clinicians, the technology developers, and the AI developers in order to get the final results and the destination. And here what you see, it looks really complicated figure, but what means this is a kind of how we will develop and master the data that we will generate throughout the project in order to understand more the risk factors and to provide that toolbox for the clinicians, which is a kind of software that you can click the risk factors, the data of the patient, and you can get the chances for getting the cancer. And with the hope that we will do so not only for patients, but also for people that seems to be healthy. The word healthy, it's really a kind of definition that we have always to understand what meaning healthy. It's control in our scientific work. Beyond, of course, as I have noted, you will hear more details about each of these features later on, but I would like to emphasize what is the role of the LUCIA project in this era. First of all, and this is the take-home message, it's to identify eligible patients. Again, many patients or many people don't know they have the disease. And this is a critical point. If everybody has the suspicion that he or she has a disease, certainly they will go to check up. But this is the main problem. If we feel healthy and we go to do sport and we do our active daily life practices, okay, without any side effects, we don't go to the doctors. Apparently, nobody will go to the clinical doctor to say, hey, I want to check, I'm healthy, right? This is not realistic, and therefore we want with these tools to identify eligible patients, namely with whom to focus our technologies and toolbox in order to further advance them. And this is a huge impact to the society, citizen, and healthcare system in Europe. And furthermore, if we identify the risk factors for lung cancer, we can enable the patient to be a partner in the decisions, not in all the decision, or at least in discussing the context of the further analysis and further diagnosis and the treatment that he or she might get in the future. Of course, while taking into account all the ethical issues and the challenges in this process. Something else that will be brought by the LUCIA project is to facilitate the administration of experimental therapy and monitoring. Though we will not apply a treatment or therapy within the project per se, identifying the risk factors and tailoring the diagnosis according to the risk factors, namely to make not only a personalized treatment, to make a personalized diagnosis, that will be one of the best outcomes of this project. Namely, to make a personalized diagnosis that will enable the clinical doctor to give a personalized treatment according to the conditions of that person. That will be a huge contribution, and that will increase the survival rate, that will decrease the side effects, and of course, that will be a huge save to the clinical system. So personalized early diagnosis will be one of the major issues that has to be done here. And hopefully, by understanding the risk factors as both confounding factors or as our target, that might also accelerate the regulatory approval for such technologies in order to get being implemented in reality. Furthermore, the LUCIA project will take the patients from their passive role during their journey in the diagnosis and the treatment into the so-called active role. I would love to see all the patients to have their active role, that once they are diagnosed or have a identification or know about their risk, then they will be active to check themselves daily, to discuss their health with the different stakeholders, so they will get more cost effectiveness and more efficient pathways for treatment. For example, even though it's not related to lung cancer, just take the case of breast cancer. In the case of breast cancer, if there is a genetic mutation called BRCA1 or BRCA2, we know that the person, the woman, might have a history, a family history that might indicate she might be at risk for getting cancer in the future. All those women that have BRCA1 and BRCA2 know that they have to do a usual checkup, and they have a little bit more incentive to do this checkup. And I hope we will have those risk factors that we will say, you have this risk factor, please be aware and be active on your health. That will be a great outcome of LUCIA project. And of course, I'm really very confident that we will reach many of these milestones because of the simple reason that really we have a great consortium with the great partners. And as you can see, we can categorize the partners to technology providers, which you can see the logos and their names here. You can see also that we will have 4 centers of clinical validation, and we have also the biological understanding also in place. And furthermore, we cannot proceed into such kind of projects without ethics and legal management, because we want really to make sure that we preserve these aspects, which are really important for equal opportunities in society. And of course, policy making and communication and dissemination partners are also part of this project, something that increase the chances for success and for bringing really high value into the field of lung cancer. With this, I would like to finish and thank you very much for the this day, wonderful day, and I wish you, everybody, to have a wonderful day and rich with a lot of knowledge and also information that will be gained later on. Thank you very much.
¿Estoy ya? Okay, thank you very much, Professor Haik, for your very interesting presentation. I'm pretty sure that now our audience, especially those that are following us online, are very much aware of our objectives here in the LUCIA project. So now we will start with the next session, which is the first session, lung cancer, a clinical perspective. We have a set of very well-known speakers right now. They are experts in their field, and they will give us much information about lung cancer, about the present, the new trends, and the future of this disease. The first speaker is Clara Freik. Clara Freik, she has studied biomedical sciences at the University of Amsterdam, followed by a master's in epidemiology at the LMU in Munich. For her master's thesis, she researched global premature deaths from cancer and at the International Agency for Research of Cancer of the World Health Organization. She is interested in the intersection of epidemiology, public health, and biomedical sciences of cancer prevention research, and is currently doing her doctoral studies at the Division of Clinical Epidemiology and Aging Research of the German Cancer Research Center, DKFZ, which is one of the partners that are working in the LUCIA project, with Professor Dr. Hermann Brenner. For her doctoral research, she's focused on methods to improve lung cancer screening and risk prediction models, working for and within the framework of the LUCIA project. So I give you the floor. Okay, thank you very much.
Thank you very much for the warm welcome. So I'll be talking about the epidemiology and lung cancer risk factors. And as John already mentioned, our team is headed by Professor Dr. Brenner, who unfortunately cannot make it today, but we are from the German Cancer Research Center. So first of all, to give an indication of the global burden of lung cancer, and then I want to go into some of the risk factors that we already know of. Of course, there's still a lot to research on this, as we already heard. And then to describe the regional variability, which also reflects the lung cancer risk factors and the differences in the prevalence, and what are some of the options and opportunities we have to improve screening and try to optimize that. So we already heard this, but lung cancer is the leading cause of cancer deaths worldwide. And so key part is that the stage of diagnosis heavily influences the treatment outcomes, and that gives us it shows us that time is really everything, and it gives us opportunities to try and reduce mortality through improving screening measures. So tobacco smoking is, of course, the leading preventable cause. It causes around 80 to 90% of lung cancer cases, and the person's individual risk is increased by 15 to 30 times. And the malignancy risk is impacted by the intensity at which a person smokes, the duration, and also since when they stopped smoking, if they did. So around, if you look at the other side of the coin, from smokers, there are around 1 in 5 that end up developing lung cancer. And it's not just the people who smoke, but also people who are exposed to secondhand smoke that have an increased risk. And we can see it's important also to look at the changes in prevalence of tobacco smoking. So we see here a map of the countries in blue are the ones who have actually managed to decrease their smoking prevalence in the last 3 decades. And the countries in red are the ones whose smoking prevalence have increased. And many of these countries are low and middle income countries who have not yet implemented these tobacco control strategies. And this, of course, also reflects then the changes in incidence and mortality that we see in lung cancer. So globally, it's still that 22% of the global population smokes tobacco, and with Europe being a region where there's still the highest prevalence, and we see differences by sex, and also that in adolescents and young adults, e-cigarette usage has increased, which also contains carcinogenic products related to lung cancer. There are also some other lung cancer risk factors, though, because as we have already heard, there are 15% of lung cancer cases that are among non-smokers, and we haven't yet been able to explain it all, but some of the research that has been ongoing has shown that there are 3 kind of different major groups. So we have environmental and occupational exposures. And this, for example, is unprocessed biomass fuels, which still around 50% of the global population is subjected to through heating and cooking. There's arsenic-contaminated water, radon and asbestos is found in construction materials, and diesel exhaust and air pollution. And then we also have the family history and other diseases. So we have, of course, genetic variants that have been found to be associated with lung cancer, as well as chronic obstructive pulmonary disease, for example, or also history of certain infections such as HIV and HPV. And then there are also, as a third category, the lifestyle factors, so related to diet and metabolic factors. And our knowledge on, our current knowledge on lung cancer risk factors have helped us also come up with different types of risk models. And these we have also, these use our knowledge of risk factors to try and predict a person's probability of developing lung cancer. And we can see here different risk factors that use just variables that can be obtained through questionnaires. So, for example, demographic information, smoking, comorbidities, and occupational exposures, as well as family history. And in our own sort of ESTER study, which is a cohort of around 10,000 participants that we have followed up over a course of 20 years in Germany, we found that these 3 different types of risk models perform the best and are actually able to predict a person's ability or probability of developing lung cancer. But there are, of course, still ways also to improve these risk models. And some of our attempts of doing this so far have involved using, for example, the microRNA score, so small RNA molecules which affect the gene expression of genes. And we have seen that adding these onto these kinds of standard risk models, we are able to predict the probability of developing lung cancer even more. And the same we have found also with certain inflammatory protein signatures that you can see here, and as well as certain methylation DNA signatures, so certain methyl groups or certain locations in the DNA in which methyl groups have bound to that affect also gene expression and a person's risk of developing lung cancer, and that has also been associated with their habit of smoking. So our future aims are also, of course, to continue in this path of trying to find ways to optimize our ability to screen people and try to maximize the benefits for people for screening by being very precise in who we select for screening and who might be better not to be screened. So this is sort of wraps up my presentation, and I'm open to any questions.
Video, no? Okay, so our next speaker is Dr. Julian Guillot. He's a specialist in respiratory medicine and intensive care. His clinical focus is on pulmonary fibrosis, interstitial lung diseases, vascular lung diseases, rare lung diseases, and radiomics-based models. His research focus is on exosomes and extracellular vesicles, biomarkers, biological functions, the diagnosis, and therapeutics. His interest focuses mostly in the artificial intelligence and radiomics, and he is an investigator in interstitial and vascular lung diseases. He is also head of clinics, respiratory diseases at the University Hospital of Liège. And his talk is on diagnosis and staging of isolated pulmonary nodule and artificial intelligence. So,
to be here for this virtually for this presentation, and sorry for not being in person in San Sebastian today. I would like to thank Vicomtech for the organization of all those presentations. So my job today is mainly to discuss the clinical approach of the lung nodules for patients at risk and not at risk of developing lung cancer. So you know the diagnosis of lung cancer is the starting point of the diagnosis of lung cancer is mainly the CT scan. We can identify lung nodules that are the first step of the lung cancer, but we can also identify the cancer at the later stage. That's why we really need a clear way to identify at the earliest stage those patients with a clear view of the screening program. So we already discussed with Clara that lung cancer is very prevalent and incident in our countries, and we really need a focus of the good patient to detect at the earliest stage. So we use mainly in clinics the two main clinical trials that have been the first starting point of the screening program in many countries. In Belgium, I have to disclose that we do not have structured screening program, but this is clearly realized by all pulmonologists around the country. So we use the National Lung Screening Program and the Nelson trial that are the best trials that have been studied for the screening program of patients suffering from lung cancer. We know that in US there is a preventive service task force that recommends annual CT for patients with more than 20 years history of smoking. And we have no evidences that this can help in the early detection and early treatment of those patients. But there is no clear view for the patient who are light smokers or with mutation in their family that can increase the global risk of suffering from lung cancer. And secondly, this is quite important to know that the exposure of the patient over the life of the patient is something that we have to take care of. And this is quite challenging for the clinicians to identify the best patient to detect at the earliest stage. Propose the screening program. Based on the previous results of the NLST and Nelson study, we know that there is a very interesting and important thing to propose to the patient that they have to benefit from the screening program. We know that we can reduce the overall mortality of lung cancer by determining at the earliest stage the occurrence of lung nodules and the prevention of the transformation of the lung nodules in lung cancer. We know that when we find the nodule at the earliest stage, we are able to propose to the patient a surgery that can cure the patient without other treatment afterwards. So the key aspect of the clinician and the key point for the clinician is the identification first of the nodule. And clinically, generally, we use CT scan, of course, but some of us are using a computer-based approach for the detection of the nodule and to help the clinicians in the detection of the nodule. So as we already discussed, the early nodule identification is our key objective for proposing the curative therapy for the patient, like surgery or stereotactic radiotherapy. We need, of course, a good expertise in the radiological department, because if your radiologists are not thoracic radiologists, you reduce the potential of identifying at the earliest stage the disease. Of course, with patients with severe disease, there are genetic artifacts that can modify the capability of the radiologist to analyze the CT, which can reduce the sensitivity of the imaging approach. We can compare imaging, and based on the morphology of the image, we can some of the time have difficulties to detect the vasculature or the tissular structure, so it's quite challenging some of the time. We use only longitudinal follow-up for the quantification and the evolution of lung nodules, but generally this is a human-based approach and not a computer-based approach, which is reducing the accuracy of the analysis. And at the end, we need the determination and of the need of biological sampling. We use PET-CT for the identification of hypermetabolic lesions. And we know that hypermetabolic lesions are at the high risk of being lung cancer in the lung. But nevertheless, for very small nodules, we are not able to use those kind of techniques. So we need something that can help in the definition of the sign that can help the clinicians to say, okay, this is probably an early lung cancer. What we use in clinics nowadays, we use the lung rats methods for the lung prediction of the malignancy. So we base first the analysis of the lung nodules on the sole size of the nodules, the number of the nodules, and the fact that the nodules can be of ground glass, partly solid or solid. And this can change the way that we propose the patient a follow-up at 3, 6, or 12 months or more. Of course, we include in this analysis, based on the Fleischner recommendation, that are the ones that are used in the daily clinics in our center. We include the epidemiology of the patient. If the patient already suffers from a cancer at time, we are more aggressive for the follow-up of the patient. But also, we know that if the patient experiences a high smoking exposure history, that we have to be more cautious with the way that we propose the follow-up of those patients. So this is a very difficult to read slide, but the main thing that we have to understand from this slide is that there have been a lot of studies focusing on the capability of AI-based model to identify lung nodules and to predict the possibility of being benign or aggressive malignant lung cancer. And this is very important for the clinicians, because we know that when we have doubt about the fact that the nodules can be or not a lung cancer, and if we do not have any longitudinal follow-up of those patients, we are some of the time in between two chairs without knowing what we have to do for this specific nodule. But if we identify that the nodule is increasing in volume or in size at least, we know that the risk of identifying a lung cancer is increasing. And based on the previous study we can find in the literature, we can identify tools that can help in the identification of the malignancy. But the real problem is that this has not been validated in clinics in prospective trials, and the sensitivity can range from 80% to 100% with a specificity from 72 to 96%, which is quite difficult to accept for the clinicians. We know that we have to deal with the risk of missing the possibility of treating the patient with a curative approach. So out of the human eyes that we can use in the daily clinics, the radiological approach from the radiologist, we use computer-aided detection for the diagnosis of lung nodules, which is the CAD approach. This is very helpful for the clinicians because this is like validation of the fact that we didn't identify any nodules, or this can help the clinicians to quantify the number of nodules and to determine whether this is there is an increasing number of nodules over the time in patients with multiple nodulopathy. The key point for the CAD is that there is a good performance, and this is very nice for identification of the lung nodules, but the positive predictive value is not sufficient based on our clinician approach to use it alone. So this has to be combined with the clinician's approach, and this can of course assist the clinicians in the evaluation of the performance of other tools. So basically what we are using for CAD is, first we run the analysis after having analyzed the CT scan. So the blue sign is identifying the nodules, and afterwards we can see whether we can quantify the volumetry and the shape of the nodule. This is the first step, at least, of the AI-based computed partners for the clinicians, but it's not enough for clinical use. Of course, at least alone. Secondly, we use the lung RAT score. The lung RAT score helps the clinicians to predict the possibility of being malignant for the lung nodules that you are identifying. We use the volumes, the tonality, the density of the nodule, and the number and the risk of the patients. And based on that, we can stratify the risk prediction of being suspicious of lung cancer. We also use another tool, which is a computer-aided tool that clinicians can help. So the volume doubling time, which is which is defined as the time required for growing nodule to double its volume, and this helps clinicians to see, okay, the volume doubling time is quite low, so the risk of being lung cancer is high. So if the nodule is increasing over the time with a very slow course, we know that the risk of being malignant is low. And we use 400 state to represent a high likelihood of malignancy for our VDT. To detect the nodules, which is, as you understood with the previous discussion, a key point for the clinicians, we use other type of reconstruction of the images of the CT. So we use the maximum intensity projection, which is called MIP, and it helps to combine all the voxels together to see better the modification of any structure in the lung. So we put everything together and project it on a sole image to help the clinicians to see the nodules. For instance, when you see the CT scan, which is the middle image, you cannot really see the nodule, but if you combine all the slide that you have upper and lower, you can see that the paramediastinal nodule, which was not identifiable before, is easily identifiable for the clinician. This helps the clinicians to see the nodule, but this does not help really for the definition of the nodule itself, but at least you can identify first, and afterwards you recheck the images to see whether you can better define the nodule and if there is speculation or infiltration or contact with the vessels or whatever you can see. Based on those kind of approach, we know that we are able now to use other tools that helps clinicians for the definitions of the nodules. And we are used to deal with radiomics in our center. We are very friendly with the radiomics because this helps the clinicians to first identify the volume, but we know that we can do this with many systems and many tools. The size first, the shape of the nodule, the intensity of the density and the texture, and the correlation between all those voxels together helps to predict the possibility of being lung cancer. And on top of this, we know that now some models and some tools are able to predict the histology of the lung nodules, but I won't go through the details of this kind of model because I guess I know that there are other talks focusing on this. But the thing that is interesting for the clinicians is to know whether the nodule is at high risk of lung cancer, to have a reliable way to quantify the nodule, but also to predict the histology of the lung nodule, because if you can predict the histology, you can go faster in any decision you have to make. For instance, if you need to perform surgery, you can go very quickly, but if you go for a stereotaxic approach because the patient cannot benefit from the nodule biopsy, for instance, you have to deal with the risk of not identifying the subclassification of the lung cancer in this nodule. So radiomics can help also in this kind of approach. From the clinical trials we can talk about, we have the Sibyl trial, and they validated the deep learning model for the prediction of being a lung cancer for those nodules. This is very important because this is the first step for the clinicians to have traceability of any kind of model that we propose based on AI approach. The results in the Sibyl trial are very nice, and they use retrospective cohort to validate their models. At the end, if we pull together all clinical trials that we can find in AI-aided diagnosis tools for lung cancer, we have a quite nice sensitivity and quite nice accuracy of all those models pulled together. But again, for the clinicians, we need a unique way to evaluate those patients and to have a reliable way to quantify and to predict the possibility of being lung cancer. So, of course, for the clinicians, we need the first to know whether we have to perform CT for those patients, but the identification is, by the way, the main issue that we have to deal with for the patient. We have to identify the nodule first. We have to have a reliable way to identify those nodules, so we cannot only based our approach on the sole expertise of the radiologist, so the CAD approach or any other approach can help in this. There is another value of other reconstruction that the normal reconstruction we use in CT scan. The size is important, so size matter for when we talk about lung nodules. The volume doubling time is very important, so quantifying the volume of the lung nodules over the time and having a reliable way to evaluate the evolution of the lung nodule over the time is very important. Lung rats can help with the prediction of being lung cancer or not, but this is one of the first steps of the approach that we can propose to our patients. There is a need for implementation or integrate those kind of models in clinics. Anyway, we need validation of those models for the identification, then the quantification and the matrix of the nodules, and then to predict the histology based on the radiomics or other AI-based approach. So I'm done with this. So thanks a lot for your time, for your consideration, and happy to answer to any question afterwards. Thank you.
siguiente, ¿no? Vale. Okay, so our next speaker, she is Dr. Monica Saiz. She is an area specialist physician at the Cruces University Hospital in the Department of Pathological Anatomy. Her specialization is in thoracic pathology, lung, pleura, and mediastinum since 2012, and in ENT and maxillofacial pathology since 2019, with active participation as a member in their corresponding committees. Her expertise is in molecular pathology, dealing since 2012 with the detection of molecular alterations in different tumors, using different methods, platforms, and devices, such as FIS, RT-PCR, etc. She's also a member of the Pathological Anatomy Transplant Group since 2012, and she has participated in different clinical trials, such as IM-Power, Histocell, AM-Canes, and publications in scientific journals. She's also an associate professor at the Department of Medical Surgical Specialties at the University of the Basque Country from 2009 to 2020. Her talk will be about the histological subtypes and molecular analysis of lung cancer. So I'll give you the floor, and thank you very much.
Okay. Yeah. Okay. No. Well, my name is Monica Saiz, and I'm going to talk about histological subtypes and molecular analysis of lung cancer. As we have already heard here in other talks, lung cancer has been the most common cancer in the world and remains the leading cause of cancer-related deaths. This type of cancer affects more men than women, and incidence increases with age, with a median age at diagnosis of 71-year-old. Okay, the majority of lung cancer patients initially present with regional or metastatic disease, and this affects their survival outcomes with poor prognosis due to the late diagnosis. Okay. In this image, I want to explain the typical patient journey from the diagnosis, the initial consultation with the clinician, diagnosis, and treatment based on type, stage, and a specific subtype of lung cancer. We, the pathologists, are in the middle of this journey because we can make the histological diagnosis of the lung cancer, and we also make the molecular subtyping of lung cancer using different methods I will explain later. Oh, sorry. Yes, first I will start with the histological subtypes of lung cancer. We have a small percentage of lung cancer, which is small cell lung carcinoma, and the other one, the biggest component of lung carcinoma is the non-small cell lung cancer. This is an heterogeneous disease that can be divided into 4 main subtypes. The most frequent is adenocarcinoma, and then we have squamous cell carcinoma and other types like large cell carcinoma, large cell neuroendocrine carcinoma, etc. There are more than 50 histological subtypes of lung cancer. Okay, so to make an accurate diagnosis, we have to take, or it's primordial to take an accurate tumor sample. And for this, clinicians have different methods, BUS, CT-guided biopsy, fine needle cytology, etc. And with these methods, pathologists have a tumor sample, a small biopsy specimen, to make the morphologic diagnosis. Okay, so in this, I don't know if you can see the images. In the first one, I don't know how to point the images. Well, in adenocarcinoma, we can see glands, okay? And another patterns of growth like micropapillary patterns, lipidic patterns. So if we can see these patterns, we can call it adenocarcinoma, okay? If we can see pearl or another keratin pearls or other signs of keratinization, we can call it squamous cell carcinoma, okay? In the middle, the image in the middle. And if we can't see any clear morphological sign of differentiation, we only can call it non-small cell lung carcinoma, and we will have to make an ancillary immunohistochemistry panel for the subclassification. Okay? We use markers like TTF1 or P40. Here in red, you have these markers. And if the TTF1 is positive, it will be adenocarcinoma. And if P40 is positive, it will be squamous cell carcinoma. Here, we have real photographs of our job, of our routine job. Okay, we have a small diagnostic sample, a bronchoscopy biopsy. Here we can see bronchial mucosa with respiratory type epithelium, and beneath the connective tissue. In the connective tissue, we can see islands of tumor cells, and there are large cells, so it's large non-small cell lung carcinoma. Okay, but we can see glands, we see no keratinization signs, so we cannot say whether it's carcinoma, squamous cell carcinoma or adenocarcinoma. We use immunohistochemistry here. We have in our institution a double panel of immunohistochemistry to determine the morphology. TTF1 we have already seen, and CK5-6. Okay, the first one, TTF1, is a nuclear staining in brown, and here we don't see it. CK5-6 is in red, it's positive, so it's squamous cell carcinoma. And the other one is another double panel with P40, nuclear and brown positive, and CK7, which will be in red, it's negative. So with this double panel, we can see it's a non-small cell lung carcinoma. Immunohistochemistry favors squamous cell carcinoma. Okay, this is the morphological algorithm to make an accurate diagnosis in non-small cell lung carcinoma. Okay, but furthermore, we have molecular subgroups to determine, and we, the pathologists, also make these determinations along with molecular biologists in the molecular board, okay? So, on the molecular laboratory, sorry. And why are these molecular subgroups so important? Well, because there are some alterations, some molecular alterations that in the personalized medicine are along with a right drug, with an effective drug against the tumor cells. So, we have a patient selection based on molecular profiling and an adequate treatment to these patients, okay? And these are the molecular alterations we already determined using different methods. And above, you can see the different treatments. So, in these guidelines on testing predictive biomarkers in non-small cell lung cancer, this is a national consensus of the Spanish Society of Pathology and the Spanish Society of Medical Oncology. In this article, we have all the biomarkers we already have to determine, not only on metastatic stage, but also some in the earliest stage, because there are some treatments after surgery, okay? For example, FGR or PD-L1, that we can use them in earlier stages other than advanced disease. Okay, so these are the essential biomarkers in this guideline, in this consensus guideline: EGFR, ALK, ROS1, BRAF, PD-L1, NTRK, RET, KRAS, and MET. Okay, PD-L1, we determine it, the PD-L1 status, using immunohistochemistry. Okay, and we see the tumor cells in brown, and we have to count them, providing a percentage of tumor cells. The other biomarkers are determined using other methods. In our institution, we use NGS, next generation sequencing, since the last year, more or less. But we have used for years other methods like as FISH or RT-PCR, etc. Okay, there are other biomarkers in progress, and perhaps we have to determine them in months or in years. Okay, like HER2, etc. Here we have, you have the other biomarkers. Now the essential biomarkers are in red. And and the last image is just to explain you our workflow in the laboratory. Okay, here we have the sample tissue. First, we have to do always the morphological diagnosis of the tumor. We have to say if it's an adenocarcinoma, an small cell carcinoma, squamous cell carcinoma, etc. Because the treatment, for example, is different. If it's a small cell carcinoma, the prognosis is poor and the treatment is completely different. Okay, so we can make this morphological diagnosis using traditional stainings like hematoxylin-eosin, and using immunohistochemistry too, as I've explained before. And then, if it's necessary, we will make a molecular subtyping of the tumors using immunohistochemistry to determine PD-L1 status, and other methods like NGS to determine all the molecular biomarkers. And thank you very much for your attention, and thank you very much to Bicontest for the opportunity of this talk and this collaboration. Thank you.
A coffee break? Vale, okay. So now we have the coffee break, the much needed coffee break. But first of all, yes, we will take a photo of all the speakers. So if you please come here and we take the photo. To the stage, yes, all the speakers, please. Those who have spoken and those that are to speak yet, all of us, please.
Bai, bai, ostras, sí, vale.
Okay, so welcome back again. Right now, we will start with another session. This is a double session on the lung cancer treatment, surgery, radiotherapy, and systemic treatment. The speakers are Dr. Unai Jimenez and Dr. Xavier Mielgo. Dr. Unai Jimenez, he has a medical degree in the University of the Basque Country. He is a thoracic surgery at the Cruces University Hospital. She's actually a staff of the thoracic surgeon in thoracic surgery department, and he's also the surgical director of thoracic surgery department in Klinika IMQ Zorrozaurre. He's specialized in lung cancer surgery and complex chest wall surgery, collaborating professor of surgery in the University of the Basque Country too, and he's a member of the editorial board of Archivos de Bronconeumonología and a researcher in the LUCIA project. Xavier Mielgo, he is also a clinical doctor. He graduated in medicine from the University of the Basque Country in 2005, completed the residency training in medical oncology at the University Hospital of Cruces in Vizcaya, and he works as a medical oncologist specialized in the treatment of lung cancer and other thoracic tumors at the University Hospital Fundación Alcorcón in Madrid. He's a resident mentor and honorary professor for the medicine degree at the Rey Juan Carlos University. He coordinates the Master of Thoracic Cavity Neoplasms at Alfonso X University in Madrid. And he teaches also 2 other thoracic oncology master's programs. He's a member of the board of directors of the Spanish Transversal Group of Uncommon Tumors and the Spanish Group for Cancer Immunobiotherapy. He is also the coordinator of NIVO-RARE study and a clinical researcher too. So we will start first with the speaker, Dr. Unai Jimenez, on the treatment for lung cancer. Thank you very much. I give you the floor.
Good morning. Thank you very much for the invitation and welcome to everybody. So I'm a surgeon and I'm going to speak about the surgical treatment of lung cancer with this first message, an optimistic vision of the lung cancer, because we have been here all the data about the surveillance, but I think that are happening really good things for the patients in lung cancer, just in the local disease, in the advanced disease, and also with this project in the prevention and early detection. So I'm not going to speak about no data of surveillance today in this presentation. Every patient lives, okay? And this is the other important message, and this the concept of your lung cancer team. The multidisciplinarity in the treatment and diagnosis of lung cancer is vital. In my opinion, as a surgeon, have no value, okay? The value, the most important value is the opinion of the team, of pathologists, medical oncologists, etc. So we have to work together, and this seems that is really easy, but it's really, really difficult because there are a lot of things in the area. The thoracic surgeons were involved in many situations and many times of the diagnosis and treatment of the disease. We can act into the wall and diaphragm, into the pleura, into the lung, airway, mediastinum, yes, with palliative intention, diagnostic intention, and also in the curative intention. So I'm going to speak today just this because the surgery is the main treatment with radical intention, okay? Sometimes alone, other times with other treatments like chemotherapy, immunotherapy, and radiation. It's a vast issue to explain how is the surgical treatment of lung cancer, but I'm going to resume with these 4 questions: who, when, what, and how. First, who. There are main concepts of the resectability and operability concepts, okay? Resectables are the tumors, and operables are the patients. So the resectability is the potential, safe, and radical resection based on preparatory studies of a tumor with a demonstrate or very likely prognostic benefit, okay? And this is the operability. Obviously, it's not the same to operate the old man, that is a heavy smoker, that have a poor lung function, that have comorbidities, that to the other is a photo of myself, the other one. Thank you. So if you are going to operate myself, obviously there is no risk there. Okay, it's very important to assure the lung function. I'm not going to explain all the criteria for assure to the value of the operability, but just to know that there are well-established guidelines with the specific lung function studies just in Europe, in American. There are also risk models to try to assure the morbidity and mortality of the lung resection. No one is good at least, but are very, very interesting for in the concept for investigation, okay, to can compare populations. The patients that are not fit enough to be operated have alternative treatments. The most important is the radiotherapy, the radiosurgery, that actually, this is a very critical conflict, the value of the radiotherapy in early stages. But in general, for patients with very poor lung functions that you can't operate, the radiosurgery, the SBRT, is indicated with good results, but only in early disease, okay? When? Okay, all the stages can be surgeon patients. In the stage 1, representing here stage 1, 2, when very small nodule without nodal involvement, not ipsilateral, not contralateral. The main treatment is surgery, upfront surgery, the first strategy, and then it could be indicated, as Xavier could speak later, the target therapy in some cases, and adjuvant with chemotherapy in tumors of more or 4 centimeters, stage 1B. In stage 2, the surgery is also upfront surgery, then is indicate the adjuvant chemotherapy. Actually, in our country, in Spain, because those can change in other countries, this indicated adjuvant immunotherapy if PD-L1 is more of 50%, and the target therapy, like in the stage 1, if is a mutation or if failure. The adjuvant radiotherapy if incomplete resection. Stage 3A, this is actually a really novelty here in this stage, because there are recent trials combining chemoimmunotherapy before surgery and then the surgery if there is no progression of the disease. This is a real revolution. 30% more or less of the patients are in this stage at the diagnosis, and actually a lot of these patients are going to be to surgery with radical intention, with this new approach. Those are Nadine 1 and 2 trials, and Checkmate 816 trial that are really news, and maybe then Dr. Miego could speak about this. In the stage 3B could be in T4 and 2 considered the neoadjuvant strategy, but this is in most of the cases not a surgical stage, okay? It's more frequent chemoradiotherapy and immunotherapy if unresectable. And the stage 4 could be indicated the surgery if it's insulated brain M1 or suprarenal isolated M1. Radical intention in selected cases I have put there because those are really a specific situation and patient that you have to speak in the multidisciplinary board and to decide which is the and future could see, because look at this, we published this paper about the neoadjuvant treatment in non-small cell lung cancer, speaking about the introduction of immunotherapy, and when we finished the article, and just appeared the results of the Checkmate 816, and we don't speak about this article in the paper, so we say, oh my God, what are we going to do? And we published, Dr. Mielgo quickly wrote another one saying that the time goes really fast and the things are changing, and this is the reality. What to do? Those are the main typical resections. Since 80s, 90s, the standard surgery have been the lobectomy, the resection of one lobe. This is has been the standard for 30, 40 years. And just to say this, that maybe in these times, in the lung cancer era, where very small nodules are diagnosing, the lobectomy could be a surgery, too much big surgery for this intention, and there are really recent trials speaking about the semectectomy versus lobectomy, the sublobar resections in small and peripheral tumors. And there are still conflicts, but could be the new standard in the next few years. There are also special techniques like this, sleeve resections with bronchial reconstruction, also with pulmonary artery reconstruction. We can do extended resection, extended to pericardium, extended to chest wall, carinal resections, diaphragm resections. With vascular chest wall, we can be really aggressive with the patients, and we can do a lot of things, but always in the multidisciplinary mode. Regardless the type of surgery, also we have to perform a complete lymphadenectomy, okay? Just N1 lymphadenectomy or N2. This is not have to be surgeon dependent, because there are well-established guidelines to how to management intraoperatively the lymph nodes, and this is also conflicting into surgical scenario. And how to do? Open surgery, minimally invasive surgery, by video-assisted thoracic surgery, by robotic, 3 holes, 2 holes, 1 hole, no holes. There are a lot of things to do. Hundreds of articles about this. Maybe the reason could be, if you can perform the surgery by minimally invasive surgery, perform it. Almost in the early stages. But the most important thing is not how do you, is to do, with quality criteria, okay? Okay, I'm surgeon, and I'm going to put a small video how we operate. This is my favorite approach with 2 holes. This is those specimen like I manipulate in there is a special bipolar cautery that we can coagulate and cut. We have like adhere to cut. Here we are dissecting the superior pulmonary vein, and now is the main truncus of the right pulmonary artery. The surgery is right surgery and right superior. So this is the ahí está. Sí, lo avanzó. Sí. Ah, perfecto. Vale.
No, ahora no funciona.
¿Se puede avanzar esto?
Sí. Con esto puedes apuntar.
Ah, vale, perfecto.
Entonces ahora, espera. A ver. Joder. No.
Vale. For the next time. For the next time. Thank you very much.
Sorry.
Good morning, everybody. Thank you for the kind presentation and echo, and thank you for the invitation to the organizers. I am Xavier Mielgo, and I am medical oncologist specialized in the treatment of lung cancer. I would like to provide an updated brief summary of the treatment, of the systemic treatment of lung cancer. First of all, I'd like to say that lung cancer treatment has undergone a deep revolution, especially in the recent years, with dozens of new indications. It's probably the solid tumor with the highest number of new indications. And I would like to explain how we do select the treatment for a patient recently diagnosed of lung cancer. We have to consider 4 important factors. First of all, the stage, if it is localized, if it is metastatic disease, the histological and molecular subtype. These are tumor-dependent factors, but there are also patient-dependent factors as performance status, that is the physical condition of the patient, and the comorbidities, that are the other diseases that the patient has. Okay, Monica introduced before the classification of the lung cancer, but I would like to say that the classification of lung cancer has been changing over time. And first of all, we treated lung cancer as a new unique disease. Later, we subclassified it in non-small cell lung cancer and small cell lung cancer. Small cell lung cancer is the subtype with the poorer prognosis and with a scarce development in the last years. And I'm going to focus this speech in non-small cell lung cancer patients that accounts for 85% of patients. Non-small cell lung cancer are divided in a practical way in non-squamous non-small cell lung cancer and squamous histology. And thanks to the better understanding of the tumor biology, we have discovered different molecular alterations that can lead to the development of the disease, and we are now in the molecular classification of lung cancer. Okay, we have to stage the patient. We use the TNM system, international system. The T component refers to the size and the local involvement of the primary tumor. The N component refers to the locoregional lymph node involvement, and the M component refers to the presence or not of metastatic disease. Okay, we can group patients according to the T, N, and M components into 4 different stages, from stage 1, the earliest stage, to the stage 4, that is the metastatic disease, and we can offer different treatments for each stage, as Unai introduced before. The greater stage, the worse prognosis. Okay, here are survival curves. Oncologists are very used to evaluate and to analyze this kind of graphics. I don't know if you have seen before, but here, when a patient is diagnosed, or when we start a new treatment, all patients are alive, and every time a patient dies, the curve drops 1 point, and we can see what's the percentage of alive patients along the time. And we can see that stage 4 has very poor prognosis, but even in earlier stages, the prognosis was not very good in lung cancer. Fortunately, we are changing the history. When we have the stage of a patient, in the earliest stages, you not said it, we can, it's enough with local treatment, mainly surgery, but beyond stage 1B with a tumor of 4 or more centimeters, we have to add systemic treatment in the context of a multimodal treatment or multidisciplinary treatment, and the goal of the treatment is curative. On the other hand, when we have in stage 4, it's metastatic, and the goal is palliative, and the treatment is mainly systemic therapy. These are the pillars of cancer treatment. We can see here the local therapies, surgery and radiotherapy, and on the other hand, systemic therapies. I'm going to group the systemic therapies in these 3 kinds of systemic treatments. First of all, the oldest one, cytotoxic chemotherapy. These are chemical agents that interfere in the cell cycle of the tumor cell and leads to the apoptosis of the tumor cell and to the death of the tumor cell. So it's like dropping bombs to the body, and it has significant toxicity. On the other hand, we have the targeted therapy. On one side, genomic-driven targeted therapy. There are several molecular alterations, oncogenic alterations that Monica explained before, that can lead intracellular signals for proliferation and survival of the tumor cells. If we block them, we can attack the tumor in a more precise way. So this is the era of the precision oncology. On the other hand, we have anti-angiogenics that can help us to normalize the vascularization of the tumor to penetrate better our systemic treatments. And on the third pillar, the systemic treatment pillar, is immunotherapy. Immunotherapy has been a major breakthrough in the treatment of cancer in the last years. The people who developed it, the checkpoint inhibitors, won a Nobel Prize, and there are different kinds of immunotherapies, the majority of them in clinical development, but nowadays in our clinical practice we use checkpoint inhibitors, CTLA-4 inhibitors and PD-L1 inhibitors. I'm going to try to explain it. Our immune system has some regulatory systems that are inhibitory signals so as to avoid self-damage. When we blockade these inhibitory signals with checkpoint inhibitors, it's like if we unleash the brake of our immune system and it finds the tumor cells as an enemy, it attacks them and kills them. So, getting to the point and talking about the treatment of lung cancer, of non-small cell lung cancer, talking about early and locally advanced non-small cell lung cancer, local treatment is determinant. And systemic therapy is complementary to improve curative outcome. The goal is curative, and when we give adjuvant, we give the systemic therapy after surgery, we call adjuvant treatment, and when we give it before surgery, we call neoadjuvant treatment. Okay, when a patient is has been resected of early stage non-small cell lung cancer tumor beyond stage 1B with at least 4 centimeters of size, we have to offer the patient adjuvant chemotherapy with a cisplatin or carboplatin doublet chemotherapy. What do we achieve with it? We achieve a gross increase of survival of 5%. And targeted therapy has also moved to the earlier stages, and if the patient has an EGFR mutation, we have to offer him adjuvant osimertinib for 3 years. This is because it's demonstrated in the ADAURA trial better progression-free survival and better overall survival with a gross increase of 10% of survival. Okay, and in a patient without EGFR or LAK mutations with a high expression of PD-L1, we have to offer adjuvant immunotherapy for 1 year nowadays with atezolizumab because it demonstrated also an increase of overall survival of almost 17%. We can also give the systemic treatment before the surgery. It has some advances, but if we see historical data, if we give neoadjuvant chemotherapy to a patient and then he undergone surgery, we obtain also a gross increase of survival of 5%, similar to the adjuvant intention. In the last years and the last months, there is a cumulative cumulative evidence of the benefit of giving chemoimmunotherapy in a neoadjuvant setting and then surgery. I'm going to point out the work of the Spanish lung cancer group with the NADIM studies because the NADIM 1 trial was the first study showing an impressive benefit of neoadjuvant chemoimmunotherapy with impressive data of pathological complete response. Here, I present the results of the NADIM-2 study, that is a phase 2 randomized trial comparing neoadjuvant chemoimmunotherapy with nivolumab versus chemotherapy alone in stage 3-1-3B patients, and 37% of patients who received neoadjuvant chemoimmunotherapy had no residual tumor after resection, no residual tumor, compared with 7% who received chemotherapy. It also correlated with a benefit in progression-free survival and overall survival. Okay, after that, other 3 phase 3 randomized studies confirmed the benefit of neoadjuvant chemoimmunotherapy. This is the first one. When I talked about it, Checkmate 816 with neoadjuvant chemoimmunotherapy with nivolumab, 6 cycles, then surgery, and it confirmed the benefit in pathological complete response, and it correlated with benefit in event-free survival or relapse-free survival. There are other 2 studies with perioperative immunotherapy, 4 cycles of chemoimmunotherapy, then surgery, and then 1 year of adjuvant immunotherapy with durvalumab or pembrolizumab with similar results. But when the stage 3 non-small cell lung cancer gets curative, with curative intent, but it's not resectable, we cannot offer surgery to this patient. We have to offer the other local therapy, that is radiotherapy, also with curative intent, concomitant with chemotherapy. And if the tumor has PD-L1 positive expression, we have to add consolidation immunotherapy with durvalumab for 1 year. You see, with this strategy, the Pacific study showed that we can improve the 5-year overall survival from 36% to 50%. That means that 1 in 2 patients are going to be alive after 5 years of treatment. Okay, and what happens with advanced disease? The treatment is mainly systemic treatment, and the goals are different. The principal goals are improve quality of life and lengthen the overall survival. And occasionally, we can offer also local treatments with palliative intent. We have 2 different contexts, patients with no molecular drivers and patients with molecular drivers. In patients without molecular drivers, it matters histological subtype and PD-L1 expression. In patients with PD-L1 expression, high expression, higher than 50% of 50%, we can offer only immunotherapy in monotherapy. It's enough. We can avoid the toxicity of the chemotherapy. But if the tumor has a PD-L1 expression less than 50%, we have to offer the combination of chemotherapy and immunotherapy. And in non-squamous cell lung cancer, we can add anti-angiogenics. We can do an induction of 2-4 cycles and then end maintenance treatment. We have up to 6 different regimens in non-squamous non-small cell lung cancer and up to 4 regimens in squamous histology. Oncologists feel like a children in a candy shop when we have to select. But the magic of immunotherapy occurs in the late tail of the survival curve. Here, there is a percentage of patients that do not progress after years of treatment and are alive after years of treatment. Maybe some of them can be cured. And what happens with patients with molecular drivers? Nowadays, we have up to 9 molecular drivers with approved targeted therapy. EGFR, LAK, ROS1, BRAF, RET, MET, NTRK, HER2, and KRAS. In the most of them, we should start with targeted therapy, and in some of them, we start with chemoimmunotherapy and then targeted therapy. What do we obtain with targeted therapy? Normally, we obtain a rapid response of the tumor and sometimes a Lazarus effect. In a few weeks, the tumor can almost disappear, and we can improve the overall survival. With all of this, we are successfully reducing the mortality of lung cancer. These are my take-home messages in early stage and locally advanced lung cancer. The local treatment is determinant for curation, but multidisciplinary management is needed beyond stage 1B and tumor of 4 or more centimeters. Systemic treatment improves the percentage of cured patients. And in advanced lung cancer, systemic therapy improves the quality of life and overall survival. In oncogenic addicted non-small cell lung cancer, generally start with targeted therapy. And in non-oncogen addicted non-small cell lung cancer, immunotherapy or chemoimmunotherapy as first-line treatment. The percentage of long survivors is increasing, and we are seeing it in our consultations. Thank you very much.
Okay. So the last talk, the last speaker will be Juan Carlos Trujillo. He graduated in medicine and surgery from the Autonomous University of Barcelona. He completed his residency in thoracic surgery at Hospital Universitari Mutua Tarrasa. He completed his PhD outcomes of VATS thymectomy in patients with myasthenia gravis without associated thymic tumor, with a result of excellent. He is currently head of the thoracic surgery department at the Hospital de la Santa Creu i Sant Pau in Barcelona. He is a medical associate professor in the Department of Surgery at the Autonomous University of Barcelona. He is also coordinator of the research project in thoracic oncology of the Spanish Society of Pneumology and Thoracic Surgery, and he is also the coordinator of the Spanish group for the study of thymic tumors of the Spanish Society of Thoracic Surgery. He is also coordinator of the Cancer Screening, Smoking Cessation and Respiratory Assessment Cassandra project, together with Dr. Luis Seijo. His talk will be about the state of the art of lung cancer screening. Thank you very much, and I give you the floor.
Good morning to everybody. First of all, I want to thank to the organizers, BioContact, and Dr. Jimenez for inviting me to this excellent workshop. It's a pleasure for me to be in this city with good weather. And I'm surgeon too, but I don't have videos. I'm really sad about that. But I'm trying to explain the state of the art of lung cancer screening. I'm going to start with the same numbers. Here, my conflict of interest and the summary of the table of contents. And I want to start with the same numbers, the same variables that we have discussed during this morning, the current situation of lung cancer. I think it's important to emphasize these numbers because I think these numbers justify projects like LUCIA. And because we have a really health problem around the world. Lung cancer is not a problem in Spain, it's a problem around the world. And this problem can be resolved with lung cancer screening. I'm not sure, but it is probably a weapon that we can offer to the patients to decrease the mortality. Here we can see the patient that we diagnose in a stage 4 or a stage 3, and here we can see here is a problem. We diagnose the patients too late. And what about the money? The money is important around the world. And here we can see that the cost that we invest in lung cancer is more than if we compare with breast cancer, with colorectal cancer, or prostate cancer. So it's not only important the mortality, it's important the money too. And why is mortality so high? Well, the problem is 80% of lung cancer are diagnosed when they have already spread. If we compare with other cancer, for example, breast cancer or prostate cancer, here is a different related to mortality. And what about primary prevention? We are talking about the risk factor is tobacco, but we are smoking less than 10 years ago. Well, we have seen in the epidemiologist's map different colors with blue and orange. This data is about the situation in Spain, and I'm not really sure if we are in blue or not. Because if we compare the results and we do this, if we compare according to the different ages, I'm a little bit worried about the age between 15 and 24-year-old, because the number of smokers is increasing in this rank of age. And I think we have to fight to do something more to decrease the number of smokers in our country, but I think it's the same around the world. The impact of screening. I want to show the 3 different trials: American trial, Nelson trial, and MILE trial. We have more trials around the world, but I think these are the most important 3 trials that we can discuss. And in this slide, we can see the impact of screening on overall mortality and lung cancer mortality. If we compare NLST, mild, they offer decreasing mortality between 7% to 20%, and according to lung cancer mortality, between 18% to 40%. I think we can criticize these trials, of course, but probably they offer information that we can use to fight against. We have a weapon to fight against lung cancer. I'm sure that it's not perfect, this kind of trials, but of course they are offering something more. And different countries are using this data to create different lung cancer screening programs. Can you see China, Hungary, Poland, Netherlands, UK, and USA? They are trying to implement different pilots and with the idea to implement a final lung cancer screening program. But European recommendation is they are following this idea to implement different trials around the world. Well, there are different recommendations. The European Parliament, they calls for the evidence that proves the positive effect on targeted lung cancer screening or mortality to be recognized. So they offer the possibility to implement different pilots around Europe to see the feasibility to start with lung cancer screening around Europe. SAPEA, Science Advice for Policy by European Academies, they also offer the possibility to start with different pilots around Europe. And the European Beating Cancer Plan, they told us that we need to do something more in 3 cancers: lung cancer, prostate cancer, and gastric cancer. So European governments, they are talking, they offer us the possibility to start with this new screening. But what about Spain? The current situation of lung cancer in Spain, it's really similar to other countries around the world. We have more patients, the incidence is increasing, and the mortality is not decreasing. So we have a problem. The mortality in lung cancer represents 72% of incidence. What we have done in Spain, well, we don't have an early detection program actually in Spain, and our government doesn't recommend the implementation. And in 2016, they decided they collect different data, and they recommended not to implement this screening. A few years after, they considered that the overdiagnosis is estimated at a maximum of 10%, but they didn't recommend the implementation. And a few weeks ago, they published another document emphasizing that it's not cost-effective option to introduce in our health system. But they opened the door to different pilot programs. We have different experiences in lung cancer screening in Spain, yes, but all of them, or most of them, are in private care. So they don't consider this data enough with enough potential to be considered to write this document. For example, in Navarra, in Valencia, and in Madrid, they have screened a total number of patients, 4,000, 8,000 and more than 1,000 patients in Madrid. They have experience, they have collected different data, but the government doesn't consider enough to start with a lung cancer screening program. This is the idea because we need more data in our country in a public health system, and this is why we created the Cassandra project. This is the key point of my presentation. I want to show this project. I'm really happy because finally we have achieved a very good experience. And Cassandra is cancer screening, smoking cessation, and respiratory assessment. We want to do another step to improve the lung cancer screening program in our country. We need to collect data. The developers, I work with Dr. Luis Eijo in Madrid. We wanted to ask different questions. Is our healthcare system ready for the implementation of lung cancer screening? Is lung cancer screening cost effective? How should we select the cases to be screened? Are we doing all we can against smoking in Spain? Well, the challenges, implementation feasibility, complementary tests that help to detect possible patients, correct selection of cases to be screened, risk modeling. We are talking about the risk model in a previous presentation, but I think it's not actually it's not possible to start with a risk model in selection because we don't have enough data. We don't have data about smokers, so we can start with different risk factors because we don't have unupdated data. Improvement of primary prevention, and all the questions we can resolve by a pilot program. And of course, we are not discussing about the effect of primary prevention. We need to work together because I think it's the most effective weapon against lung cancer. This is the first time that all the societies are fighting with the same idea to improve the diagnosis and to trying to decrease the numbers in mortality, and we are working together with the patients too. I think it's important to talk with them because they are the principal actor. And the objectives: a patient-centered screening. We are not centered the screening in cancer. We are trying to promoting respiratory health. It's a good opportunity to do that. A screening beyond cancer. If we do a CT scan, for example, we can detect other diseases like COPD, for example, or fibrosis, other diseases related to tobacco. We recommend to do with a low-dose CT scan, but we add an spirometry and a blood test because it's important to create more data to improve the lung cancer screening in the future. We created a unique protocol with a scientific committee with different scientists with a huge experience in lung cancer screening. And the current status of Cassandra project, well, we are trying to fight against government, but we created a unique database. It's important to collect the same data around our country. We conduct a quality control and audit, an external audit, an homogeneous reading of the image, because there are different programs to read the images, and it's important to read with the same system, and a blood database because it's important for the future. Biomarkers will be crucial. So the key points: unanimity. It's the first time, like I said before, it's the first time that all the scientists agree with the idea to fight against and to create a lung cancer screening program, homogenized to a single database, external CRO, a unique software, a scenario, public health system. Finally, we have the scenario that the government want to study the feasibility. A good geographic representation, because we need patients. The inequity is around the world, and in Spain we have the same problem. And depending on the city that you live, you have the more feasible or not to access to a CT scan or to access to the oncologist or to the surgeon. Tools for the future: blood database, risk models. Probably, if we start with this database, we create or we collect all the data to create a specific risk model in Spain, because I think it's not possible to create a unique risk model in Europe. I think, and I always say that each country has to create his own risk model and improve primary prevention. Actually, we have more than 40 hospitals around our country, and not only hospital, primary care units too, and we created an inclusion criteria to decide which hospitals can participate or not, and you can see the different communities and the number of hospitals that they participate. We are really happy because we have 16 communities involved in the project. We created a web page trying to make a diffusion to the society, to be part of the society too, to show the updates that we achieve. And the future of lung cancer screening. I think this kind of project, this kind of pilots offer us the possibility to improve the lung cancer screening. The idea of LUCIA to improve the diagnosis and to decrease the mortality in the future. The risk modeling, of course, we need to select the population better. It's not enough only with the age and tobacco addiction. I think we need to identify other risk factors, and we need to be sure that it is possible to collect this data in our country. We decided to use the same variables that the USPSTF used in the States. They created in 2013, they updated in 2021, and they consider to be screened a patient depending on the age and depending on the tobacco consumption, and they offer a 65% sensitivity, but actually we know it is not enough. If we compare with other systems, risk modeling, for example, PLCO, we have different risk modeling. I think that when you have a lot of options, we don't have the perfect risk modeling, so this is the idea, but the future, probably, they offer more significantly sensitive, but they can screen patients older, had more comorbidities and shorter life expectancy. So we have an example of PLCO, but the future probably is to create risk modeling based on other criteria. Here, an example, biomarkers, to improve the selection criteria, to manage the indeterminate nodules, to characterize size of small nodules, and to determine, for example, the low-dose CT scan intervals. Here, an example, autoantibodies. We are working in our country actually with two projects, Refine and Define, and the idea is to use this test to detect 7 autoantibodies in a blood test that our body produces in response to the presence of 7 tumor antigens, and with the idea to, for example, if we detect a nodule in the CT scan, to be sure if we have to repeat in 6 months or in 1 year, depending on the results of the test. So we can conclude that lung cancer continues to be a serious problem around the world. Prevention alone is not the solution. Screening should be a potential weapon against lung cancer. Evidence in favor of screening is increasing. European pronunciation, I think a country like Spain has to follow the European recommendations. Spain has a previous experience in different autonomous communities. Let's learn from it. Screening has room for improvement, but let it pour. Risk modeling, biomarkers are part of the future, and Cassandra as an example of pilot project. Thank you.
Okay, so now we have time for some questions and answers to start the round table. So if someone has any questions, comments, I don't know if someone from the audience, maybe in the, gente del vídeo que está en casa puede, online, no, okay. So maybe I have a question. Okay. Okay.
Basically, basically what you said is that Spain is not really following the last results or recommendation of the European Union, and there's some reluctancy. It has to do possibly with the cost for the health system and so on. What will we need to demonstrate exactly that this is effective and that it should be implemented really? The last part, no?
Well, I think it's a little bit complicated to compare, because I think the key point is to compare the cost effective in lung cancer that we have to use in lung cancer screening, comparing with the money that we use for treatment, for example. If we compare, they are considering that we have the cost is 30,000 per ABAC. And if you compare, for example, with the treatments that Dr. Mielgo presented us, if you compare, I think the prevention, it's an option. But the future, Spain actually, we need to continue fighting against government, but I think they are opening the door to create a pilot project, and I think it's good for us, but I'm not sure about the future. I'm not sure.
And one last question: what is the size or estimated size of the pilot project? Number of participants?
50,000 people.
Another question for Dr. Trujillo. Is primary care involved in the Cassandra project, and how is the involvement of this community?
The idea with this project is to consider that primary care is crucial of course, to indicate which patient has to be screened or not. And we have 40 hospitals, but each one has at least one primary care unit related with the project. We consider that it's crucial to improve or to implement a future lung cancer screening program is to have primary care units involved in the project.
Thank you.
Yeah, more questions there?
So I just wanted to ask about your data in general. So post this project, is that data going to be accessible to other research projects to work? Or we have a huge issue in UK making the data accessible to researchers, and the gatekeepers want to keep it locked, and that really restricts us on what we can do. So is there the opportunity within your country to be more open and share?
Yeah, actually, at the moment, we need to start, but of course we are open to collaborate with other projects, because I think it's crucial to learn a little bit more. I think we have to learn about UK pilot, for example, and actually we have different data. I think it's not the same. In Spain, we have a different health system, but I think it's important to collaborate. Of course, we are open to collaborate with you. Yeah.
Yeah, there's a question there, in the back.
They make me stand up. Okay. Good, I'll do that. I want to thank you guys for these presentations. Being not related to any clinics, this is really very, very interesting information that you've handed over. I've never sort of seen it put together in such a nice picture. So thank you very much for doing that. That's my first comment. I have a question. So you say the sort of the criteria for inclusion into screening programs is based on age and is based on whether someone smoked a certain amount of pack years. On the side of the genetics, people are talking a lot about polygenic risk scores and so on. So what is your opinion on the inclusion of polygenic risk scores into such a as an inclusion criterion for screening, for example?
It's a criteria to be considered in the future, but actually, for example, in Spain, it's impossible to include patients according to the polygenetic. I think in the future, if we have a database of more than 50,000 people, and we have a blood test of each one, I think we can create different criteria that actually it's impossible. For example, in Spain, we don't know exactly the number of smokers that we have. So it's impossible to consider genetic, actually. But of course, in the future, I think it's has to be considered.
More questions? Okay.
Thank you for the presentations. What I wanted to ask, you mentioned that a good, maybe it was mentioned that good treatment would be early diagnostics or screening. So we are working here in the LUCIA project on technologies. We're working on different risk factors, as Ivo mentioned. As physicians, what is your look? What do you think will be more good in the real world, in the clinic? Aiming towards more of identifying risk factors, or aiming towards neurotechnology or things like that? What do you think will be better? Open question.
Yeah, it's open. Yeah. I start, but I think they can answer too. I think both, because technology. I think the risk factors and trying to identify, they are trying to work in the same way. We need to improve together technology and risk factors. As a clinician, of course, I need to improve technology, but I want to operate more cases. Then if we improve the other way, I think in the future we can use more technology for more patients. This is my opinion.
I agree with Juan Carlos. For me, it's more important the new technologies, more than the technology is more important for me than the risk factors for improve in the real life. I think that could put not more information, but more useful for the patients. Sorry for that, Ivan, because your risk modeling.
I agree with both of you, but I think that the better knowledge of the risk factors will be important if we can avoid them. If we cannot avoid them, that's no way. So nowadays, it's very important to develop the technology to detect earlier the lung cancer. It's my point of view.
I have one more. We are also on the project looking a little bit about the environment. So do you have some already, I don't know, is there already knowledge about the environment is changing all the time and the effect? Is there already studies showing different aspects regarding lung cancer and the effect of what's going on in the world globally in changes in the environment? Does it affect?
Yeah, actually, we have a group in Spain that they are working specifically in radon, for example, and they are studying the difference between the. I think they started in the beginning of this century, and they continue studying, and I think that we have more information, and they are trying to see the changes in radon, but. I think in other risk factors it's not possible actually, but for example they are studying the changes in these risk factors.
Yes, also with the asbestosis and amianto, I think that we have information about the industries, and here in the Basque Country, for example, we have had a lot of factories with a lot of naval construction, so this is directly relation with the ambient population. So we have experience with the asbestos here in the Basque Country, I think in Spain in general. So, but no study. I think that is really difficult to apply in the real life. How to extract the data. I think that this had been a problem, no, for all of us in this project to structure the data about how we live, how we eat, where we live, and the factor associated with this. So it's really challenging this question, and it's really challenging for the project in general to know this. So.
Well, this is a general question. I don't know who should be answering this. Probably it's a difficult answer. So when we look at the molecular subtypes and when we look at in detail and what's going on in lung cancer, we see that there are some gaps, and sometimes they are huge, like 30%, 40% of unknown origin or something like that. You show the numbers. LUCIA is about prevention, but also about understanding. So if we have some cases, our question is what we should be looking at to discover this. I mean, to discover or to explain these cancers. So I think that there are a few advances in small cell lung cancer in trying to figure out molecular subtypes and so on. So what with the technologies, we will explain the technologies later on. But if you could provide some clues of what we should be looking at to discover something else in terms of data types or information, or I don't know. Maybe it's an open question. And okay.
So you are talking about the lung cancer not related to tobacco, or?
No, I'm not talking about the risk factors. It's when you already have some cancer and there are some types that are not well explained, their biology, their origin, their molecular subtyping, and so on. Where do we start?
One of the principal problems of the diagnosis of the subtype of lung cancer is the tissue. The tissue usually is very little, and it's difficult to find. Sometimes it's difficult to make a good diagnosis and to make a good histologic subclassification and a molecular classification. Sometimes we don't have enough tissue to make a correct molecular diagnosis in a high percentage of patients. And that's a very big problem for us, and I think that sometimes some rare kind of tumors or some specific histologies are related to some specific molecular alterations also. Yeah.
And possibly other things that I see is that since the tumors are usually detected later on, probably they have already some history of evolution, genetic alteration, and so on. So they are maybe so maybe it's more difficult to classify where do they come from or what were their.
You want to ask something?
When we use the NGS to identify the molecular subtyping of the tumors, we have found that there are some molecular alterations not targeted with no drug, with no effective drug, but there are alterations. For example, P53, it's nearly always, or in most cases of lung cancer, alterated or overexpressed, but there is no targeted therapy to this molecular. And there are a lot of alterations in NGS that we know, but with no therapy. So perhaps in these tumors, we will find some other molecular alterations, but the thing is that in NGS, it's a platform in which we find only specific biomarkers. In bigger platforms, with more genes, we will probably find more molecular alterations with target or not target therapy, but we will probably find. But these biomarkers or these alterations, when they are not related with targeted therapy, there is no clinical way to use them. So in the practice in the routine medicine practice is not, they are not used.
Hi. Oh, that's too low. So I have a question regarding when you spoke about the histology, that sometimes you don't have enough sample to classify it. I was wondering, what do you think is the role of like liquid biopsy, for example? Because if you don't have like enough tissue to make like a classification, there are like some studies that are correlating like the amount of tumor cell that you can find in blood or like the DNA. Do you think like this could help making a better like classification?
Yes, it's an important question. The liquid biopsy is suffering also a deep development nowadays. It has different utilities. We can use it to find these molecular alterations that we can target with targeted therapy, and but it has some advantages and some disadvantages. When we find a positive alteration in liquid biopsy, we can believe that is this real, and we can treat the patient. But there can be also false negative results. And in those situations, I recommend to continue looking for the alteration in the tissue. Okay. Another advantages is that normally the lung cancer and other tumors are heterogeneous. It can recognize the heterogeneity of the tumor with a liquid biopsy, and we are also using liquid biopsy to see the resistance mechanisms to the targeted therapies, because it's easier to take only the blood of the patients than making a biopsy. And we can repeat it several times without big morbidity. And the other utility of the liquid biopsy is the minimal residual disease, specifically in early stages. It's prognostic, and it can help us to decide if we have to give more treatment or longer treatment. It's under development. Yes.
Thank you.
More questions? Yes. More questions? Yeah. I want there. And the last one.
Hi. Thank you everyone for the presentations. This is also an open question to whoever has the answer. So it's pretty much established by everyone that early diagnosis is key and very important, and the cost of saving someone's life who has late-stage cancer, the surgery, the CT scans, the time element of queues in hospitals and wait times to be seen by a doctor or a cancer specialist or an oncologist or whomever has a lot of financial impact. And we understand that early diagnosis is relatively much cheaper. However, in LUCIA, some of the main concepts that we're working towards are different technology products for early stage diagnosis. The question is, how would it be possible to convince the person who is responsible for the budget on the hospital side for purchasing these products? Because there's no financial return on something that may save someone's life. We know surgery does save someone's life. We know a scan will show that someone does have cancer. But if you're doing widescreen testing on people that may or may not have cancer, how do you justify that expense? And how can we make sure that these concepts, or how can you help to make sure that these products enter the market and hopefully take over some of the stress that's given from the heavy-hitting expenses like surgery in the field?
Well, I think it's really difficult to convince inside the hospitals about a project like LUCIA, for example. Because if I compare with Cassandra, for example, I have to convince the director of the hospital that I want to start a trial to improve the diagnosis and decreasing the mortality, but always we finish the conversation talking about the money. And it's a little bit difficult because at the beginning you have to give money to start with different CT scans, and the government. For example, here in Spain we have 17 communities, and you have your own money. And of course, if you decide to give this money to the CT scan, you have to convince that this project is so important for the hospitals that you have to continue with this idea. And I think it's not. We are always thinking about the money, and we need the money now. If you talk about the money in the future, it's not important for the directors. It is my opinion. It is sad, but it is a real wall. But I think we need to fight. For example, one possibility to convince the directors, talking about the CT scans, for example, actually, in clinical practice, we are doing a screening. Pneumologists, for example, they decide to do a CT scan year by year as COPD patients, for example. But they are using a complete CT scan with all the technology, and they are using a CT scan that probably you can use a low-dose CT scan, and you can use the other CT scan for other patients. This is a possibility to convince the directors, and this is a model that we are using, that you have to recanalize, to do another ways to not to reduce the need of the money. But I think it's a little bit difficult, the possibility.
Okay, so I think that was the last one, wasn't it? Yeah, okay. So we can, okay, yes, last one. The very last one.
Thank you. I was thinking about the implementation of lung cancer screening programs at national level, and all this discussion about cost efficiency of these programs, and also the mandate or the recommendation from the European Commission and our national authorities, and how to make these screening programs targeted. So, taking into account that we cannot target screening for polygenetic risk scores because we do not have that information actually, but on the other hand, we already have vast amounts of information about air pollution, environmental exposure, and it's open data actually, and we have the technology to link it to specific populations, not at patient level, but maybe at neighborhood level. And that's something that is quite surprising for me, that we have heard in all the presentations, the huge advances in molecular, in genetics, and in therapies, but almost none of those advances are related to how to integrate this information in the lung cancer screening programs. I'd like to raise this point because for me it's a surprise. Haven't heard nothing about that. By the way, my background is technologist, I'm not a clinician. And I would like to hear the opinion of the panelists about this point. Why are we commonly neglecting, at least from is my perception, neglecting this maybe potentially useful information that could make these cancer screening programs more effective and cost effective? Thank you.
Adriano?
Yes, exactly.
Totally agree. I think we have the same problem with other risk factors. I think we are not neglecting the information. This data is crucial, I think so. And for example, last year, the Spanish Society dedicated the year to improve the quality of the air, and I think we have data, actually different cities or different communities in Spain, we have identified the potential risk to develop not only lung cancer, other cancers too, related with the environment. So we are not neglecting. I think we don't have enough data actually to start, because it's really difficult to possibly we create an inequity if we decide to start, for example, we start in Barcelona, the lung cancer screening, because we have identified the environment is crucial to detect the cancer, lung cancer here. This inequity, probably we don't have enough information to start with this idea. But of course, I totally agree with you.
Okay, so thank you very much. This was the last one. And now Alba will moderate the next session.
Thank you. Thank you, Neko. Ladies and gentlemen, good morning, everyone. Thank you, everyone, for coming to this workshop. And after the exciting session where we have learned a lot about the clinical matters of lung cancer, we will proceed with the workshop's second session that is dedicated to exploring new approaches and solutions, innovative solutions for understanding and detecting lung cancer. We have assembled a distinguished panel of experts who will share the insights, experiences, and breakthroughs in detection methods, molecular analysis methods, and the application of artificial intelligence in lung cancer. The first two speakers are Dr. Joab Broza and Dr. Osnat Selatavor from Technion University, Dr. Broza, and from Nanos Medical Company, Dr. Selatavor. And first, Dr. Joab Broza is a senior research fellow and adjunct faculty of the Department of Chemical Engineering of the Technion Israel Institute of Technology. He's the head of the biological and analytical team, and his current research includes the identification of disease and infectious agents, biomarkers, and hazardous materials through volatile chemical analysis. And also, Dr. Broza serves as editorial board member for both Nature's Scientific Report and MDPI's Chemosensor journals. And he is also the project manager of LUCIA project. And Dr. Osnat Selatavor has a PhD in molecular biology, and since there, more than 20 years of experience in developing complex, innovative, multidisciplinary products in the medical diagnostic field. And after working at several companies such as Compugen, Given Imaging, and Medtronic, in the last 3 years, she established Nanos Medical and served as the company's CSO. Sorry. So, Dr. Broza, the floor is yours.
Thank you very much. Okay, I want to start by thanking our host for this workshop and for the later 2 days we still have here for our meeting. Both Vicomtech and Biocurses for the very nice organization here. So thank you for the introduction, Alba. So I'm Yoav, and I'm going to talk about the sensing solution for lung cancer screening and diagnostics in the aspect of the LUCIA project, of course. I want to give you some information or some background on the Technion, maybe those online, those here that are not familiar. So actually, the Technion is an engineering university. It's one of the top 100 universities worldwide, according to the Shanghai ranking. In our group, the LNBD group, or Laboratory for Nanomaterial-Based Devices, which is headed by Professor Hayek, we are focusing on different fields, spanning from engineering, chemistry, all the way to applicable medicine. We are doing both basic research and, as I mentioned, applicable research. We focus on volatile organic compounds as biomarkers, as communication molecules, and then in parallel develop different sensor device for identification, detection, and analysis of those molecules. So, when we are talking about volatile organic compounds, what are these folks actually? We're talking about small chemical molecules. You can see here an example. Just a moment. Okay, you can see here an example for two molecules like benzene and pentan. These evokes are chemical molecules that have high tendency to evaporate in atmospheric conditions, and they are mainly the process of different metabolic pathways in the body, but we can also find them outside, in the environment, in different man-made processes. So it's important to identify what is the real source of these chemicals when we find them. In recent years, we can find a new term or new type of omics. We call it volatolomics or volatilomics, depending who is defining it. And I guess most of you know things like genomics, proteomics, metabolomics, which is the study of the entire metabolites in an organism. So actually, volatolomics is kind of a subsection of this metabolomics. It's the volatile fraction of metabolites that can be studied. As you can see here on the right, this is the concept that we are working in this specific field, and you can look at this from the outer circle inwards. As I mentioned, some of these molecules are coming from different conditions, different processes within the body, different metabolic processes from different organs. So we can find these molecules coming from the lungs or from the colon, for example. And the next step is, okay, we need to measure them. So how do we do that? We take a sample. So we can take different samples from different sources, different bodily fluids, as we call it. It could be urine sample or blood sample, but it could also be skin sample and breath sample. And once we have the sample, now we need to analyze it. So we have the common methods or gold standards like mass spectrometry that we are working with, but we can also develop new technologies like different sensors, what we are doing in the Technion. So we develop different sensors, and then eventually we can reach the diagnostic stage or screening, and then eventually determine if we are if it's a sample from a sick person or from a healthy person. So this is the concept. As I mentioned, we are working a lot on technology and development of different sensors. So when we're talking about sensors, we have two main approaches that you can see here on the slide. So the first one, in the upper pane, you can see is the selective sensing. And selective sensing is really, I would say, straightforward. You say, okay, I develop a specific sensor for a specific molecule. I detect only this molecule, and I can measure it according to time. And then I can define some kind of a threshold. And I say, okay, if you have up to this threshold, you're considered healthy. And if you have above it, you're considered sick, or vice versa. This is a very good approach, but it's sometimes problematic because, first of all, you need to have a specific biomarker. And as you probably know, in many cases, you cannot find a specific biomarker for a specific disease. It's the body is very complex. There are different things going on, and it's part of the reason why it's harder. So if we know what we are looking for, and if we have a specific biomarker, it's a good strategy. On the other hand, we can go to what we call cross-reactive sensing. And in this case, we have many different sensors. Each sensor can react with different molecule, and each molecule can react with different sensors. It's similarly to the olfactory system, actually. So what we have here is many responses based on the sensors that we are using, and then we can use different pattern recognition methods, machine learning, and so on, to classify the data now between, for this example, healthy and sick people. So what we do in the LNBD group, we take different nanomaterials like metal nanoparticles, silicon nanowire, carbon nanotubes, and so on. And we integrate them and develop different sensors like the chip you see on the right, which include 8 different sensors that was developed in a
previous EU project, and this chip you see here is about this size, okay? And we can integrate it in a portable device and use it as a way for identification, for detection of different diseases. Sorry. So what we do, or what we're aiming in the LUCIA project, so we have two main aims from the Technion side. The first is the wide spectrum biomarker multi-use sensing patch. This is actually started in a previous EU project called APatch, and we're now taking it to the next level. And actually what we're talking here is a patch that we can stick on the skin. Okay, it's not so big, it's something like this. Again, if you look at here, this is the size of the patch. And the idea here is that it's a patch with no battery, and it's very thin, and you can put it on the skin, and it will be powered by NFC technology, okay? I guess most of you pay with your phone today or your smartwatch or whatever. So this is the same concept. You can put your smartphone or tablet, and it will power the sensors, and it will also give a measurement. So I can put this patch at T0 in the clinic, and then afterwards I can go home, measure it after half an hour, measure it after 24 hours, doesn't matter. And then I have different intervals of measurement of these sensors from compounds emitting from the skin and accumulating between the skin and the sensor patch itself. So this is the first technology, and the second technology, already mentioned in the morning, is what we call SPOCE, or spectrum radio on card, and this idea is actually a graphene-based sensors that are printed on paper. So it's paper-based sensors, and we can do layers of the sensors, and by this we can achieve kind of a chromatographic-like separation. And what you can see on the figure here, this was done on cell lines in the lab, so it's cancerous cell lines and non-cancerous cell lines. But the idea is that you can separate these samples using this technology. And in the LUCIA project, we intend to do it sorry, in blood samples. So finally, for the technology from the side of the Technion, I illustrated it here in this figure. So we have the patients or the controls provided by our collaborators here, clinical collaborators. We can stick a patch on the skin, and this will be measured, as I mentioned, by either a smartphone or a tablet, and through a dedicated app that our collaborators here from Imoda will help us, and we already started the development of this software. And on the other side, we have a routine blood sample. Again, will be taken in the clinics, and then moving into the SPOCE device, which is actually composed of sensors we are building in the Technion, together with the device that we are, again, developing with our partners, again, in LUCIA, Pronat Medical. And all of this, both of this, will go into AI analysis, again, with the help of Vicomtech to do so. And this will eventually go into the LUCIA toolbox, as you can see here, together with other aspects or other data sources that are intended for the LUCIA toolbox, and hopefully provide a useful tool for the clinicians. That's it for me. Thank you very much.
First of all, thank you very much for inviting me to introduce Nanos Medical. Okay, this is the most complicated part to push the right button. Okay, so in Nanos Medical, we develop a device, an innovative device that will enable disease screening, early detection, and monitoring, and Also that it will be made as easy as breathing. And we heard during all this morning how much important it is to screen and early detect diseases in general and lung cancer specifically, because the early diagnosis currently in lung cancer is only below 20%, and this is actually the key for efficient treatment. Without early diagnosis, all the treatments that we have, they are very limited in their efficacy. And the problem or the limitation currently is that most of the devices and tools that are suitable for, that are used for screening and early diagnosis, most of them are invasive, are unpleasant to patients. They're associated with high cost. We discussed this for actually all the lectures were just an introduction to my lecture. They're expensive. They require special operators to operate them, also contributing to the expense and the lack of accessibility. And they are not suitable for routine diagnosis screening, and also not for very tight monitoring of the patients during treatment. So breath actually has a huge potential. First, because it's very accessible and very simple to collect. But as Yoav explained, it's also very rich in volatile biomarkers that reflect the metabolism and the disease state of our body. And also this happens in very early stage of the disease. So breath is a very important matrix that our body release. And what is very unique in our technology that actually is based on the sensors that were developed in the Technion is that our device, that innovative device that we develop, is a very small handheld. It is designed to be operated by anyone, not only by specialists. So every nurse in every primary care and everyone in the future at home will be able to use this device. It's all the design of the usability of it is designed for this. And therefore, it's enabling a very cost-effective diagnosis. So the system is based on 3 parts. First is the smart chemiresistor sensor array that is located inside the device. As I said, it is based on the technology that was developed in the Technion. This is a bio-inspired sensor array. So it does not identify molecule by molecule. It is well known that different diseases have different smell. This smell is based on patterns of volatile compounds. And actually, our sensors are responding to the patterns of volatile molecules by electric signals. This is very similar to what happens in our olfactory system. Biomarkers are interacting with many different olfactory receptors, and the nerves send electric signals to our brain. Our brain doesn't know which molecule arrives to the olfactory receptors. He just gets a pattern of electric signals. Now the brain knows to associate patterns of signals with different smells. First time we smell cinnamon, someone tells us this is cinnamon, and the brain knows to associate a certain pattern with a certain smell. Exactly like that, we have these sensors that create patterns of responses. The second part of our system is the device itself, contains the electronic board that we developed for very high resolution and high frequency measurement, very accurate measurement of the resistance of the sensors. So we measure the resistance of the sensors over time during the exposure, and we have very accurate patterns, and then we collect all these response patterns in a clinical study after exposing the sensors for diseased patients and non-diseased patients. And then comes the third very important part of the system, is the algorithms that we develop, the classification algorithms that are based on machine learning algorithms that know to detect patterns of signals that are associated with non-disease and patterns of signals that are associated with disease. And the actual test itself is a few minutes test. First, we have the a breath collection unit, which is disposable and also was designed to collect the breath fraction, which is the most concentrated with the volatile biomarkers. This is also a very unique design of a unit. It is disposable, so no safety issues are connected. Then the breath sample, which was collected, is exposed to the sensors which are here in a replaceable unit that will enable the use of stable sensors and can be replaced easily, so no calibration is required, which is the process. So the breath sample is exposed to the sensors, they are measured with the electronic board inside this small device, and then the algorithms are crunching the data and will give us the diagnosis, of course, or what is the risk of you having now a disease or not. Specifically in lung cancer, we have the opportunity in this consortium to develop a very robust algorithm based on the accessibility of so many subjects in the clinical study that is planned, which is a great opportunity for us. So all this process will take a few minutes, and it's, as I said, is very accurate, simple to use, and very affordable. And to complement with other tests, we really hope that we will bring a lot of improvement to the lung cancer management field, and will enable early diagnosis. And also very important, since we heard about the so many treatments that are available for lung cancer, and also their limited efficacy, this tool, which is, as I said, the usability of it also can be suitable for home use and primary care use. The treated patients can be monitored very tightly in order to know which treatment is working very early on, because currently, as I know, I'm not a clinician, but as I know, mainly PET-CT or very costly and radiative imaging is used for monitoring these patients, and you cannot do them only once in several months. So such a tool can enable tight monitoring and to know if a certain treatment doesn't work, and maybe change, personalize the treatment, replace treatment early on in the process. So it's not only diagnosis, but also other utilities are very important. Thank you very much.
Thank you, Osnat and Yoav. Next speaker is Jonathan Sleeman. Dr. Jonathan Sleeman earned his PhD in 1992 from the University of Cambridge. After working on his postdoc, he was appointed Deputy Director of the Institute for Toxicology and Genetics in 2003. In 2008, he took up the Franz Bohard professor position at the University of Heidelberg in Germany. He is the executive director of the School of Translational Medicine in Mannheim, and internationally, Dr. Sleeman has coordinated significant projects And his research focus centers on unraveling the cellular and molecular mechanisms of metastasis. Dr. Sliman, wherever you want.
Thank you very much for this kind introduction. Cancer is a genetic disease, and lung cancer is no exception. Lung cancer develops when there are changes in gene expression or gene activity. So when we think about risk factors, we need to think about the genetics. Risk factors may act intracellularly to change the expression or activity of genes. They may also act on the extracellular milieu around cells and stimulate other factors in that milieu to then trigger transformation, the development of cancer. So in LUCIA, what we're aiming to do is to identify risk factors, but then what we have to do is to link those risk factors with genetic changes. And at one level, that will be correlative. We'll see a correlation between changes in gene expression or activity that are associated with risk factor. But the important thing is that we go beyond just correlation, that we go into the area of causality. And that's something else which you want to address in the LUCIA project, and that's where we come in. We being Jan Supek from Barcelona in Spain, Yuval Shaked from the Technion, and myself in Heidelberg. So our task is to go beyond the correlative and to go into the functional. We want to look at the risk factors that are identified in LUCIA, and particularly focusing on genetic risk factors for lung cancer development, and show or to determine whether or not these genetic changes have a functional impact on the development of lung cancer development. And of course, once one's done that, one can then start to think about diagnostics and therapy. So we also want to investigate, for example, the potential of such genetic changes as therapeutic targets. So how can we go about this? Well, we need to go back to some of the fundamentals, the genetics of lung cancer, as well as the cell biology. We've already seen some of these data before from previous presenters. There are different types of lung cancer. The major classification is non-small cell lung carcinoma. There's, of course, then obviously small lung cell carcinoma. And non-small cell lung carcinoma can be subdivided into various categories. The major one being lung adenocarcinoma, LUAC, and also, of course, squamous cell lung carcinomas. So if you think about the genetics, LUAD has basically been the most intensively studied, and we know that there are at least 10 drivers and 26 tumor suppressor genes. So what do we mean by drivers and tumor suppressor genes? These are terms which have been already talked about this morning. So drivers are genes which, then they become activated, will stimulate the proliferation of cells. So when we get permanent turning on of these genes, that will stimulate the uncontrolled proliferation of cells, which is typical of cancer. Tumor suppressor genes, as their name suggests, are genes whose function is to suppress the development of cancer. So if you inactivate those genes, then you will essentially take the brakes off and again allow cells to control in an uncontrolled manner. So we have these two kind of complementary genetic changes which occur during the development of cancer. So we've got multiple genes which are involved in this process. As I said, the understanding of this process is better understood in the context of LUAD. The other forms of lung cancer are being investigated, but perhaps less well understood. So if we want to look at the causality of additional genes in this process, we need to move into a model which allows us to mimic these multiple genetic changes, this polygenic background to lung cancer. And so that's one of the basic things which we're aiming to establish in the LUCIA project, the ability to look at multiple genetic changes all at once. What about the cell biology? The cell biology of lung cancer is also quite complicated. If we look at the proximal to the distal, from the mouth to the alveoli, there are multiple cells lining the trachea and the alveoli, which can be transformed and lead to the development of lung cancer. So until comparatively recently, it was thought that for LUAD, the major target for transformation was so-called AT2 cells. These are cells which fill the alveoli, which is where gas exchange occurs within the lung. In terms of squamous cell carcinoma, basal cells were thought to be the cell of origin. And in terms of small cell lung carcinoma, pulmonary neuroendocrine cells were thought to be the target. Recent mouse genetics studies have suggested that the situation is rather more complicated. So in mice, what you can do is you can use genetic engineering to change the DNA, which encodes key genes. So you change the genes to mimic what goes on in human cancer. And you can also mark specific cell subpopulations within the lung and follow what happens. That allows you then to track what is the cell of origin. So the outcome of these kind of studies suggests that the situation is rather complicated because there is considerable plasticity in terms of cell identity within the lung. And one type of cell can be converted into another. And this probably reflects the fact that our airway is continually being exposed to toxic insults. We're breathing in nasty things all of the time. And there needs to be repair mechanisms which allow all of these different cells to be repopulated to allow them to carry out their key functions within the lung. So we need a model which also reflects this kind of plasticity. In addition, we know that there are key microenvironmental factors which can determine also the type of lung cancer. So, for example, take club cells, a particular type of cell within the trachea. If these express particular driving mutations, they often also require inflammation in order to become fully transformed. So this is, again, an example of how the extracellular microenvironment can impact on transformation. So our ideal model should also allow us maybe to mimic these kind of aspects. Now, mouse genetics are good and have been very instrumental, but in terms of polygenic basis for lung cancer, it makes it quite difficult to model within a reasonable time frame and within a reasonable financial frame. So really we need to be thinking about in vitro models, using cultured cells in order to be able to model these kind of effects. And that's rather difficult because if you take your lung and you take these cells out and try to cultivate them, you can do that for a short length of time, but they eventually die off rather quickly, which limits the amount of things you can do to enable you to look at the underlying genetics and cell biology. So, based on that, we've decided to employ immortalized alveolar epithelial cells, so-called HBEC3-KT cells. So these are alveolar epithelial cells which have been immortalized. That means they will continue to grow in culture indefinitely, and they've had particular genes introduced, CDK4 and H-TERT, which immortalizes them. This is a model which was established by Jerry Shea in Texas. Oh, no, in Dallas, sorry. And he is collaborating with us in the development of these models within the LUCIA project. So we've been able to obtain the cells from him, and he's given us support in terms of establishing them here in Europe. So key things about these cells is that they have been extensively characterized, and they express key markers which allow us to define what kind of cells they are. So they express markers for basal cells, for club cells, and for AT1 and AT2 cells. So that means we can model, essentially in this culture system, most of the major subtypes of lung cancer. They're not positive for markers for neuroendocrine cells, so they may be less useful for small cell lung carcinoma, but nevertheless we can cover a lot of the basic lung cancer biology. And if you put these cells into particular matrices, which you can see at the bottom right here, these cells will grow and will form pseudo-alveoli and structures which are very reminiscent of the of the lung architecture as kind of organoids, which reflects their underlying plasticity and ability to recapitulate a lot of what's going on in the normal lung. And if you look at the top left, you can see kind of the characteristics of the cells growing in culture. They're basically plastic and give rise to multiple cell types as they grow. So again, recapitulating this plastic effect. So in terms of using these cells for transformation and looking at lung cancer formation, what Jerry Shea's done is introduce some of the key drivers and loss of function of tumor suppressor genes into these cells. For example, loss of P53, gain of KRAS mutations, and also ectopic c-MYC expression. And what you can then do is to use so-called soft agar anchorage-independent assays as a surrogate for transformation, the ability to form lung cancer. So basically, you take the cells, you suspend them in soft agar, and if they are transformed, they will then grow out as a colony. And that's what you can see here in the bottom picture. So if you then progressively introduce these changes, which I've just described, you can see that the number of colonies and also the size increases as a function of these genetic changes. If you put these cells into experimental animals, what you can also see is that under particular circumstances, these cells can give rise to specific subtypes of lung cancer. So they can recapitulate what's going on in the human organism and give rise to adenosquamous carcinomas, large cell carcinomas, etc. So they have this kind of spectrum of different subtypes as well in vivo. If you then compare their transformation abilities and do that through genomics, you can look at genes which are associated with high tumorigenicity and then use that information to map back onto human data. And what you can see is that gene expression signatures which you find in these highly transformed cells correlates with poor prognosis in these human data sets. So again, what you get out of these models allows you then to go back to the human situation. And finally, you can also use these cells in order to be able to test novel pharmaceuticals. This is an example here looking at EGFR inhibitors. What you can see in the top, these little black dots, which you see, these are soft agar assays. Each little black dot is a colony that's growing. If you treat with the inhibitor, you can completely suppress the outgrowth of these colonies. So again, this is a model for allowing you to investigate pharmacological intervention. So our strategy in the LUCIA project is to take the genes or the genes which are associated with risk, which come out of the work of other partners, and then to use these within the HBEC3 cells on the background of the genes which have already been changed within the different sublines of the HBEC3. So then allows us to start to model these polygenetic changes which underlie risk and which underlie the development of lung cancer. So we will test the ability of these genes to have an effect, firstly in soft agar transformation assays, and then we will characterize the cells. And what I want to point out here, we will do this not only on the single cell level, but we'll also use CRISPR-Cas12 technology. This is genetic engineering technology, which allows us to modulate the expression and activity of multiple genes all at the same time. Okay, so once we've then done this in vitro work, we then want to go in vivo and look to see what happens when we genetically change these cells. Can they then form lung cancer in vivo, and what kind of phenotype do these tumors have? And once we've then got verified functional risk factors, we can then do the classical transcriptional profiling, looking at cells with and without these changes, and then do pathway analysis to identify potential candidates, which we can then use for pharmacological intervention, which again we can use our in vitro system for that. So that basically describes the kind of pathway which we're aiming to develop and establish within the LUCIA project. And we hope that it will serve as a paradigm for other investigations beyond the life of the LUCIA project in order to identify genes which can be used both for diagnostics and therapeutics for the benefit of lung cancer patients. Thank you.
Thank you, Dr. Sleeman. Next one is Dr. Ivo Good, that he's the director of the Centro Nacional of Análisis Genómico based in Barcelona, one of the largest genome sequencing centers in Europe. He is the principal investigator of several ongoing EU-funded projects and participates in large international projects and consortia, such as the International Cancer Genome Consortium, the International Human Epigenome Consortium, the International Rare Disease Research Consortium, and the Human Cell Atlas. He's author of more than 400 research papers with an age index bigger than 120, 11 reviews and 12 book chapters, and cited over 88,000 times. He's inventor of 26 patents and the founder of 4 biotech startups and serves on numerous international advisor boards. So please, Dr. Ivo.
Thank you very much. Why are you clapping? You haven't said anything yet. Okay, thanks a lot for the invitation to come and speak. I was handed this title, so this sort of created a little bit of a problem for me because I basically had to make all the slides from zero. But it's a topic that I've been thinking about for a very, very long time, and so I'm actually very happy to have this title. On the other hand, I don't have a hell of a lot of time, so I'm going to stay very, very basic with things. And I know that not everyone in the room is the absolute specialist in genomics and so on. Now, I'll start out with the central dogma of molecular biology, which is sort of what underlies this concept of multi-omic analysis. And that is that basically you have DNA, gets transcribed into mRNA, gets translated into protein. But this is not a one-to-one process. This is not that every DNA sequence that you have goes its way to a protein. It's cell type specific, it's very complicated, it's highly regulated and so on. So I'll put it to this level here. So if we have a genome, we have something that sort of tells a cell what it actually is, which is the epigenome, what is switched on inside, which genes are actually expressed and so on. Each cell has the same genome in it with 22,000 genes, but the epigenome itself has 28 million positions, CPGs, cytosines next to guanosines that can be methylated or not, and each one of those can be a sort of a switch to say whether the gene that's coming behind it is actually meant to be expressed or not, and then you have the transcriptome that comes out of that. Cell types, it's very, very interesting what Jonathan was just telling us, because I'm going to tell you something even more amazing based on this. So thousands of cell types in the body, each one slightly different, each one of them capable of expressing maybe 4,000, 6,000 different transcripts. Then when you get to the translation level, proteome, post-translational modifications, huge numbers of proteins that potentially could be inside each one of these cells. And then, amazingly enough, when you look at the metabolome, we're actually not very good at measuring the metabolome. We measure hundreds of components that are sort of the result of this cascade of things going on. A problem that lies underneath this is the dynamic range of the things. So the genome, 2 copies per cell, simple. Okay, either you have a T, a C, or you have 2 T's or 2 A's at a given position. So it's not very dynamic. Epigenome, you get 3 values because you have two alleles. You can either be both alleles not methylated, 1 allele methylated, both alleles methylated. So 3 situations that you have to deal with. However, when you get to the transcriptome, the dynamic range gets huge. So you can be 1,000 copies of a certain transcript present in one cell. So you can have very, very big differences going there. And then when you get to the proteome and you look at serum, 12 orders of magnitude difference in expression of these proteins. And metabolome, no one really knows, because we're talking about 100 that we're able to detect, and we don't really know what the range actually is. Now, in the last 20 years, we've been fortunate. We've been given NGS technologies. I won't go into how they work, next generation sequencing technologies. I'll tell you, they are in principle digital readout devices. So like your CD player, we are measuring digital information, not analog information like in your old records that sort of spin around, which means that to probe this, it's just a question of how much money you stick into the experiment. So how long do you keep sequencing to how deep can you actually go to probe this? When you look at the other omics, proteomics, metabolomics, you have devices such as mass spectrometers or NMR machines. These are analog reading devices. Analog reading devices give you maybe 2 to 3 orders of magnitude in dynamic. So you can measure something that is 1,000 times less concentrated than the most concentrated thing inside your sample, but after that, it's finished. Then you're in the noise of the experiment. There are tricks to get around it. You can use sort of chromatography to split things apart, and you use your dynamic a little bit better in each one of the windows that you have. But this is sort of the problem you're working against. Now, in multi-omic studies, usually the biggest problem is that people don't work on the same tissue. So usually they are comparing protein analysis done on a biopsy with RNA analysis done in circulation and so on. So it's not the same thing. They don't really compare the same levels with each other. A problem that they have is that the data isn't sort of complete, so you don't have the complete view of everything, so correlating things with each other is complicated. It's also an issue of the kinetics of things, so you can have compounds that are very low in concentration that are very important, so enzymes that just are there for a very, very short moment, but sort of transform things going from one situation to another. And as I said, you know, different analytical approaches, digital and analog. Now, you said this already, but I'm going to say it again. Now, correlation, just because two things are moving in the same direction does not mean that they actually are causally related to each other. And I'll give you an example. I don't have the graphs anymore, but I had these about 10 years ago. And that is, if you look in the Alsace in France, the birth rate between 1985 and 2005 was increasing like that. It was going up. And at the same time, the number of nesting storks in the Alsace was also going up. Okay, if you believe the correlation has something to do with this, you obviously believe in this. But I think we all know that that is not the case. So correlation is not causation. So you always have to keep that in mind. Now, how do you then start correlating things when they are not sort of optimally put together? So across the field of genetics, there are quite a number of really, really smart statisticians that have been working technologies or analytical methods, how to actually put multi-omic studies together. And I'll start off with this one here, which is which comes out of the Jonathan Marchini lab, is tensor decomposition. Essentially what it is a sort of a clustering method where you put all of the data in and you look to sort of you basically get these matrices that are sparsely loaded, and then you work off these. So this is a really very clever method. It was used to put genetics together with gene expression. A little bit before that, there was an approach that was developed by Steve Horvath that is weighted gene co-expression network analysis, and this has been translated into methods that sort of are multi-omic. Now, what this in principle did it built the networks that we currently work with. So it sort of brings things together that really are related to each other. So genes that are expressed and that sort of correlate with each other, brings them together by its likelihood. And what people have been using this for in multi-omic analysis is that they've then taken multiple omics levels. So they've taken, for example, the transcriptomics level, and they've done a WGCNA, which is this thing here. And they've taken the proteomics level, and they do the same thing. They do WGCNA. And then what they do is they bring the two analyses together, and they look for the modules that essentially relate to each other. So this is one way that you can do a multi-omic analysis. And then very recently out of the Stegle lab, we have this, MOFA technique, multi-omics factor analysis, which really goes the whole way. And essentially the key bit in this over the tensor decomposition is that there is an imputation step inside this. So the data itself is actually sparse, so there are bits missing, and what you do is you sort of guess what the actual value for that data point is, and you enrich it that way and bring it into a framework. So very, very nice. Okay, so what is our problem? So the problem is we analyze things in bulk. So bulk means different cell types together. And essentially, because each cell type is doing something slightly different, you just have this composite of things. Now, people have been trying to do cell type deconvolution. So you go in and you try to figure out how many different cell types are actually in my mixture, and you try to pull them apart and see what they are actually doing. What is going on inside each one of the groups of cells? Now, this in principle works as long as you don't look too far. So there are two different ways of doing it. One is supervised. That is, if you know what the cell types actually look like in terms of their profiles, you can sort of pull it apart and say I have 30% of these, 10% of these, 5% of these, and so on. That works reasonably well. If it's unsupervised, then usually it's quite a mess. It doesn't really tell you how many you have. It shows you your first few that you can have, and that's it. We did this on a data set for which we had epigenome data and transcriptomics data, and the result was really very disappointing. We didn't get the same numbers and nothing correlated with nothing. So it's not something that really solves the problem. However, in the last decade, 5, 6, 7 years, we've seen an explosion of techniques that actually allow us to do what I'm sort of going to get at, and that is single-cell techniques. So in single-cell techniques, we have technologies that allow us to profile sparsely transcriptomes in individual cells. And we can do this thousands of times in parallel in the same sample. We can also get an epigenetic readout, for example, by single-cell attack. That shows us what is actually accessible in the genome and what not. And then the key bit is that we have two techniques here. One is called multi-ohm, which does multiple techniques in one individual cell. And that ability to do multiple techniques in one cell then allows you to correlate between, for example, an expression profile, a transcriptomic profile, and an epigenetic profile. So that then allows you to bring this together and then you really use this resolution. On top of it, you can actually do something and then translate this back to spatial readouts. So I'll show you here about nearly 400,000 cells. About 200,000 of these are only single-cell RNA sequencing, 50,000 single-cell attack, 50,000 multi-ohm. And what you can see is that you can then nicely sort of project them over each other and enrich one by the other. But having said that, each cell actually is only giving you about 10% of what is actually happening in the cell. But if you take all of the cells that are in close proximity, you can sort of generate a synthetic complete profile of a transcriptome or of an epigenome and get really good information on that. On top of it, you can actually then use that. So this then means you've actually identified the actual cells, so you can see what they are, you give them an identity, and then you can take them into a spatial context, so into a histological section, and essentially within spots that have something like 5 to 10 cells underneath them, you can use this to deconvolute which cells you actually have underneath this and get a very nice resolution out of this. Okay. So, and you can do this on the highest level. So we have level 1 annotation that basically just pulls apart T cells, B cells, and so on. And then you can go down into the individual groups, go to a level 2 annotation. And in the case of the tonsil that we've done here, we ended up with 137 distinct immune cell types. So that's your number of 8 or whatever it was, 12 or something like that is probably very low. So the reality is it's always going to be a lot a lot higher, and probably if you look at your goblet cells, there's not one goblet cell, there's probably X, I don't know how many. And each one of them could be causal, or the one cell that actually then is the one that grows out into the tumor. Okay, and then the result coming off this, and you can use the information in the expression, where you see, for example, between the T follicular helper cells and the non-T follicular helper cells, you see a difference in expression. For example, for this PRDM1, the same on the level of accessibility. Whereas, for example, this one here, also the same on the level of accessibility. This is BCL6, but opposite in expression, and it essentially gives you a readout that says there is in the ATAC-seq a T follicular helper cell specific enhancer that is sort of controlling BCL6 expression. So I'm not going to explain all the technical stuff that goes into this, but essentially these single cell techniques are a very nice leg up for the application of multi-omic analysis and so on. Okay, we have a lot of smart kids who work on this, so it's fun to work with them, and thanks a lot for your attention. 2 seconds left.
Thank you, Dr. Good. For the last presentation of this session, we have 2 speakers. The first one is Dr. Ivan Macia, that holds a PhD in computer science and artificial intelligence for the University of the Basque Country. Since 2003, he works for Bicontech, and since 2014, he's director of the Digital Health and Biomedical Technologies Department, leading a group of around 35 researchers. He's also principal investigator of the Digital Health Research Group of BioGipuzkoa Research Institute. He has more than 20 years of experience in research in the area of medical image analysis, computer-aided diagnostics, data analytics, and artificial intelligence in the biomedical sector. On the other part, Dr. Kiko Núñez holds a PhD in learning healthcare systems focused on benchmarking AI-based predictive models for toxicity, onset, and severity, and real-world data-driven hypothesis generation. He's currently working as a senior health data scientist and senior project manager of the Innovation and Data Analysis Unit of Virgen Macarena University Hospital in Seville, Spain. And his work has resulted in around 100 contributions to scientific journals and congresses. So I think that Ivan Macia, you're first. Yeah, so the floor is yours.
And okay, hello again. So, okay. So I wanted to make an introduction, because we don't have so much time, about what kind of predictive models we could use in LUCIA, and a little bit about the huge data landscape, data and analytics landscape that we have in this project, without entering too much detail in advanced architectures in artificial intelligence and so on. So the first thing, if we think about models and mathematical and computational models, the simplest thing we can find is knowledge-based models. So it's based on a explicit modeling of knowledge or experience. And of course, you have also more simpler or more advanced models, depending on how you want to model knowledge or experience. So the simplest thing is what we will call rule-based systems. So also, I'm talking, of course, of digital systems, no? So how to translate experience and knowledge into computer systems. So for example, for clinical guidelines or other kind of experience, basically what you do is to construct a set of rules, and this is the simplest thing you can do. This is, for example, how we translate clinical guidelines into the decision support system. Getting into more advanced systems, we have ontology support, creating ontologies for digitally representing knowledge, or doing things like more advanced things like knowledge graphs, which have the advantage that can be processed by computerized means and algorithms. Okay. So statistical models, these are very well known. they try to adjust parameters of a statistical parametrical function to fit some data, and here multivariable data, either for classification into a set of classes or for regression to estimate the value of a variable in the future. Okay, so these are the most well-known algorithms for classification or regression, which are typically used in clinical practice, for example. Okay, when you want to estimate risk. An example, for example, multivariate linear Cox regression, logistic regression. They are typically based, these models for risk are based on observational studies with large cohorts where risk factor data is collected, usually through questionnaires, okay, plus possibly some additional information on variables, on demographics, or some biomarkers. Some of these models are typically simplified into calculators for risk prediction and easy adoption for clinicians. Okay. So, when we move to machine learning and the AI world, why would you use machine learning and all this? So, first, machine learning was developed quite a long time ago. It had also an evolution, but basically from the 80s and the most recent algorithm in the 90s and so on. The simplest, well, basically, they develop mathematical representation of data. They try to untangle the variables that are the relationship between variables within your dataset by iteratively adjusting the parameters of a complex function, some complex function that usually you cannot visually represent. And this is why they are kind of black boxes. Okay. So this iterative adjustment of parameters is called what we call training of machine learning models. Okay. So the simplest are similar to statistical models, and you could say some of them are even statistical models, but they allow for more complex models representing nonlinear functions. So this is a very important quality because sometimes the relationship between variables are nonlinear. So if you really want to represent something more complex, and you don't have explicit, very clear hypothesis of the relationship, maybe you need some of these models. Another quality is that, well, they can handle naturally a very large number of input variables and a number of output classes. Okay. And they do it without so many a priori hypotheses. If you want to deal with unstructured data in machine learning, you can do it, but probably you will need preprocessing. That is, you will need to take your input data, process it, and convert it into variables that can be directly input into a machine learning model. So this preprocessing work with images or other kind of unstructured data is typically costly and depends much on your knowledge or experience on how to do this. For example, you want to do image analysis to extract some mathematical features or calculations that you input to the machine learning model. Typically, you can see that also that nonlinearity in some of the classifications. For example, you see the third model, random forest or decision tree. You can see that they can classify two classes with a nonlinear function. Okay. This is super simple to show when you have two variable to class classification, but when things get complicated, you have many classes, and basically you have multiple dimensions that can span to even 100 or thousands. So you cannot represent visually a 100-dimensional space, for example. So this is where things get more black boxes. But still, the algorithms work. And then we have the era of deep learning models, which is a subclass of machine learning models, and this is where things get really dark, but also get really powerful. Okay, so they are advanced machine learning models based on large generation neural networks. They are exceptionally good in dealing with unstructured data or large complex data. But you need a lot of information to train them. Well, they can naturally deal with images, natural language, signals, time series, multiple molecular data, and with combinations of these, okay, with more advanced architectures. And they have the ability to extract the relevant features, patterns of the data, without so many a priori hypotheses, and perform classification and prediction end to end. You can see this in the example below, where the typical classification problem of a convolutional neural network inputs an image, just the image, and outputs whether this is a zebra, this is a horse, or this is a dog. This is one of the simplest models. But you can get deeper into the details and analysis of images or any other kind of information, or even combination of those. So, and of course, they can handle naturally heterogeneous data. They are very powerful, but they have the problem that are not so explainable, which, as compared, for example, with the statistical models, or even with some of the machine learning models. So things get complicated. They can do more complicated things, but they are less explainable and probably more difficult to validate also. So when we have LUCIA and its data model landscape, for understanding LC, we can see that it's really quite complex, but we can more or less divide our data sources and models we use into different aspects to cover the whole process of understanding lung cancer. So first, what we would like to do, and what we discover and assess risk factors in population and predict risk. Okay, so these are risk prediction models. Then, as Jonathan said, well, we have to establish a link between and a causality between the risk factors and the biology. So you need to do a detailed analysis of risk factors, causality, and transformation potential, linking the risk factors to biology. Then we have, of course, understanding the biology, how it works, and all the details, understanding molecular, cellular, immune change leading to disease. And then the disease is detected. It presents itself with some histological, molecular types, even at the staging and so on. And then we can, in this, study disease phenotypes and develop early precision diagnostics. So probably there is also intermediate. I don't mean they are whole separated steps, yeah, but more or less this is something we can say. If we go to the first part, data sources we can use are questionnaires. When you have a cohort, for example, you can use questionnaires to collect all this data, sensor electronic health records, open data sources we can do, and basically the kind of models you can develop is statistical or machine learning models on risk factors, which are the most common. And something we can do is electronic health record models for incident prediction. There is a recent literature that you can predict, for example, the risk of one cancer within 1 year with only based on diagnosis and treatments in the electronic health record. Why? They don't explain the actual reason, but there's something in coding that is telling you that there's probably some hidden cases or some incident cases already within 1 year or so. But they don't explain the whole history of where they come from. Sample size can be huge because these are population-based data. And the data depth is not, when I say data depth, it's how many details we have, how many variables, how many information, and to which extent. And the data quality is usually poor unless they are cohort studies, but still questionnaire-based assessment also has some data quality issues. Talking about questionnaires, this is where you need to study in detail. This is where cohorts are more relevant to study risk factors. Of course, you have bibliography, and you have also existing databases, and of course, then you get experimental, and this is the typical data or the typical information you use for your. If we can use some models, these are basically knowledge models. We have the AI models who try to explain regional differences in these factors and why they appear here and this population on that. Okay. And of course, we have the understanding of the cellular processes and so on, but they don't translate so much into computational models, but experimental models. Sample size in cohorts is smaller. Data depth is shallow because you are collecting, for example, questionnaire data, so the number of variables is quite limited, and the data quality is better than in population-based databases. When we go to the biology, and Ivo has explained it very well, the data landscape we have there, so I will not get more into that. And well, the sample size is more limited because these studies are very costly. So, well, there are already some very large cohorts, but for multiomics, for example, analysis, the sample size is small. The data depth is depends, but it could be. I should put three stars there maybe. And the data quality is usually better because they are very carefully selected data and analysis and so on in this kind of biological analysis. And then we go to disease. In the disease, either you have core data or you have real-world data. Okay. The sample size is usually more limited. The data sources clinical data, cohorts, biobank samples. Then we have imaging and pathology because the disease already is diagnosed. Okay. And then you can have some genomic analysis, some NGS, depending on the subtypes. The problem is that if you want to make analysis of one subtype of another, then the data set is more reduced. Sample size for real-world data is medium size. For if you really go into a specific cohort, it's smaller. The data depth, you can get a lot of information from every patient, and the quality depends on your system. And finally, I wanted to make just one. So we have a lot of information, a lot of models, and what we can do to merge these different pieces and try to build something better. Okay, so there are what we call strategies for data model integration. For data integration, the simplest one is just to integrate data sets. For model integration, what we have is early integration and fusion or late integration and fusion. Very simple early integration and fusion is, okay, you have a model, you take all the different input data, for example, risk factors, you have some genomic variants, you have some any other information, put it at the input of a machine learning model, for example, and then you build a model mixing all this. Well, this is more or less simple. It works, but not all the data has the same meaning and not all the same origin and so on. So it's not always the best strategy. Late integration, for example, develop models for different aspects or different types of data, and then aggregate them with a decision rule, with another machine learning model, depends on the complexity, and try to provide an estimate, for example, of risk by combining different approaches. And finally, we could talk about integrating knowledge and integrating machine learning just to guide the models and so on. Okay, so trying to fix different pieces. Okay, so this is all from my side, and thank you very much.
Hello, good afternoon. I think I'm the last one, so I will try to be brief, and you will be happier, hopefully. So, let me check the buttons, okay, perfect. I will touch upon three simple, maybe not so simple points during my talk. I will go through very briefly because it has already been said today, which are the lung cancer-related risk factors already reported in the literature. I will talk a little bit about predictive models for lung cancer risk that are already being used en route in practice. So it's not only research, but the ones that are being currently used in some countries, and which might be the LUCIA approach for the lung cancer risk factors modeling. So in the bibliography, we can find for the general population several risk factors already said by the previous speakers. So from the genetic point of view, we have already gone in depth about that. The exposure to tobacco smoke. Also diet and alcohol, which surprisingly haven't heard almost anything today, but it is there in the literature. Also previous diseases that led to chronic inflammation that could progress in lung cancer. Ionizing radiation, it's also another source or another risk factor related to lung cancer. And occupational exposures, of course, like asbestos, metal, silica, diesel, air pollution, exposure, as well, like particle matters in air, and also estrogen is related in the literature like another risk factors. But this was for the general population, mainly smokers, because of the studies were focusing on them. But for non-smokers, if you can see it, there is not very much differences, at least what has been reported in the literature. Yes, we have genetic, second-hand tobacco smoke in this case, but also lifestyle factors like diet and alcohol, previous respiratory diseases, leading to chronic inflammation, potentially leading to lung cancer, occupational exposures, viral infections as well, air pollution and estrogen. But, and this is something that I'd like to highlight, there are also social disparities related that have been correlated. I'm not sure at which point this is correlation or causality, but at least they are cited in the literature. We are noticing more cases in younger women than in men in general. There is also an ethnic bias. It seems that a black population are more diagnosed with lung cancer than white. Also, we have differences in diagnosis urban population versus rural, mainly because of exposure to pollutants. Regarding incomes, there are also, in this case, higher incomes areas have a higher number of diagnoses, but with better prognosis because they are diagnosed earlier, and also related to the insurance, health insurance that the population has. Usually public insurance has a late diagnosis compared to private, and this is related to the incomes, obviously. And also, depending on the healthcare provider that the population has assigned, if they have already a lung cancer screening program, so in case that they get diagnosed, they have more chances to survive more time. And these are social disparities and could be also considered as risk factors. And also bringing again the discussion that we had at the end of the previous panel about potential inequities that could cause the inclusion, for instance, of environmental exposures in specific areas. My question for discussion is, well, you are considering that this could contribute or lead to an inequity, but if we can prove that these are risk factors that can put specific populations in higher risk, from my point of view, we are not doing a good work if we are treating them as the same way as other populations that maybe, for instance, rural population that are wealthy. So I don't know. I think that there is room for discussion on that topic. So coming back again to the presentation, we are facing challenges, real challenges for developing these risk models in lung cancer because we are dealing with heterogeneous domains. So it's clinical, genetic, lifestyle, occupational, environmental, social, and maybe some more that we don't know yet, and in the future we'll see. Also, we have a mix of longitudinal, static, time-dependent, seasonal, etc. So they are variables that cannot be equally treated, processed by the model. So we need to deal with this complexity as well. And the information that we have available, retrospective information that are coming from real world, is usually not standardized, which aggregates a little bit more of complexity of the analysis, and low quality because this information was not collected for research purposes. So it was collected for taking decisions by a human, not by a machine. So these are the main challenges that we are facing in LUCIA and all the community that are trying to advance the modeling of risk factors. So the second point that I wanted to touch upon is about the predictive models for lung cancer risk. The first one that I would like to comment on, and even though this has been already referred in a previous presentation, is the one that is derived from the NLST trial in the United States, but focusing on the model itself, it's a logistic regression with a single variable, which are the screening results based on the OncoRats, lung rats score in low-dose CT scans images. They binarized the outcomes of these low-dose CTs, whether they were positive or negative. They made 3 CTs in 1 year, and then it stratified the outcome and the risk in 4 years, which was the risk of developing or at least getting a diagnosis of lung cancer. And they achieved that area under the curve of 0.76. And this was in 2019, the publication. So the next one is the one that is currently being used in the UK, which is based on a case control study. And the model, from the technical point of view, is a multivariable logistic regression, which is still classic statistics, let's say. The population involved, it was cases and controls, age and sex matched, and they included as risk factors smoking duration, so they are focusing on smokers. If there were any prior diagnosis of pneumonia, which we have seen before, it's a chronic inflammatory disease. Also occupational exposures, prior diagnosis of malignant tumor, and family history of lung cancer. And the endpoint that they were looking at was absolute risk in 5 years. And they achieved that performance with 0.71 AUC. So considerations from these two. I know that today have been said that also in China and Poland and a couple more countries, they are implementing these lung cancer screening programs. By the time I did this presentation, I didn't find any results in the literature. I will be very grateful if somebody could hand those to me. But well, the final considerations about this, they have been applied, these 2 predictive models, in US and UK only. So it's very specific, white population, with they have lots of biases due to this. And the models, I was saying before, this is classic statistics, so there's not artificial intelligence anywhere. So it's very, there's a good point that from that is that it could be explained very well to everybody. So these models can foster trustworthiness in the community. It's easy to grasp why they are predicting that. But on the other hand, we feel that it might be lacking some power that AI could provide. So this is a trade-off that we need to live with, and we need to figure out how to get the best from both sides. The population, as I was saying before, non-smokers were not included in the cohorts, and they are mostly white and Caucasian. Okay. And regarding risk factors that are being implemented in here, they are missing genetic, polygenetic risk scores, as we were discussing before. Environmental are only dealt with the exposure to asbestos in the UK case, and that's it. But nothing about social determinants. And I want to stress on this because it's very important that we are aware that we already have sheer amounts of open data and social determinants. We have the Eurostat data source, which is available for research, and you can even apply for the micro data. It's population level data, geolocated at sometimes a neighborhood, postal code, census area, but that information can be linked to specific populations, and they can help to improve the outcomes, the performance of these risk factors, and could also help us to develop more targeted lung cancer screening programs, more cost efficiency. We are talking about money during this morning and afternoon. It's a very big concern because the low-dose CTs are expensive, and governments want to invest in something that is cost effective. And this definitely could help us in that regards. And that's Yes, sorry. I was forgetting what we're planning to do in LUCIA. So for phase 1, yes, we are planning a two-phases approach linked to what Ivan was saying before. And for phase 1, we are using only EHR data, retrospective data, and we plan to develop a benchmark of classic models and machine learning and deep learning models. We will put together a cohort of around 500,000 population from Spain, Belgium, and Latvia, and with hopefully a 1 to 10 ratio of cases and controls. The candidates' risk factors at this phase will be mainly clinical, they're derived from the EHR, and we will go into the absolute risk of developing lung cancer in 1 year. And we will measure with the area under the curve, the root mean square error, and the R-square metric. In a second phase, we will add open georeferencing data that I was commenting on from Eurostat and also from national sources in Spain. We will apply the same methodology for the models. We will benchmark and see what works better, the population the same, and what we will add, hopefully, is the same in phase 1, but lifestyle, alcohol, smoking, body mass index, environmental, pollution and radon, and social determinants. And we will measure it the same way. So the challenges that we will face specifically in LUCIA is that we will be dealing with real-world data sources. So we will need to devote extensive resources for curation and pre-processing. We plan to deploy a federated infrastructure because we have the GDPR that some might say that they are burdening the development. I say that they are guaranteeing the rights of everybody when you are developing artificial intelligence tools. The population, we will also have some biases, obviously, because of we will have first imbalance of samples, expected imbalance of samples from providers. We don't have the same number of samples from Latvia than from Belgium and from Spain. And the risk factors, what we plan to do, and this is something that is on the hands of Ivan mainly, I guess, is to link the outcomes of these models to what is happening at the omics level in a deep phenotyping study. And that's it. Thank you.
Well, as we are a bit delayed, I will open the questions to the audience, but please, we don't have that much time, but we have time for 1 or 2 questions. Anyone? Don't be shy. Well, I will do it otherwise. As Professor Haig mentioned in the beginning, the objective of LUCIA is to implement the technologies that we have presented in the clinical practice. But what problems or what obstacles are we going to overcome to implement this in the clinical practice? What do you think are the main obstacles to implement the technologies we have presented in the clinical practice? Maybe clinical or any technological partner or whoever wants. Well.
Okay, so since I have proposed that, I have to provide my answer, and with the hope that further discussion can be done by the clinical partners. Apparently, all of the devices, the breath analyzer and the skin analyzer, are still in the research and development stage, even though they are quite advanced. So we have here 2 issues: the scale-up issue, which we solve. And the other issue is the ethical issue that we have to provide in order to bring, for example, the skin sample onto the skin analysis. So what happens there in terms of the clinical centers, in terms of the ethics? This is one challenge, namely how the clinical centers will digest these devices. The other device by Nanos Medical, it's advanced stage device, which you breathe in, even though it's not invasive. You need to put your mouth on it. it's important to know how you will manage the data which you collect from that. This is a small challenge, but I think this is resolvable in case the clinical studies will be done under the umbrella of research and development. So again, what are the standards that are put by every clinical centers in order to accept these solutions? This is one of the challenges, but I think with a little bit of flexibility, we can, and things can be done.
Thank you for your insights. So, Dr. Macia?
Well, there are several aspects to clinical adoption, maybe you can confirm. Well, first we need to develop, so in our case we need data, which is difficulty in access data and high quality data and so on. I think it's quite relevant, either it is from the consortium, external, whatever. So data for the questions we need to answer. So that's one point. Second, from the technological point of view, people think about, for example, treatments, and it's easy to see that treatments really have to have a process of development, approval, and validation, but technology does too. For example, we have seen with nanos, comes from a previous research from Technion, which means that there is it takes several years to take some technology to develop it and take it to the market. For example, with our AI technologies, it happens the same. Maybe the times could be shorter, but still we need to collect data, make some proof, make in silico validation, then collect prospective data, make an observational study to see that this is working, try to implement it in clinical practice. So this takes years also, even if they are digital models. And one last thing we haven't commented probably because there was no time to make yet another little session on that is that the prospective study of LUCIA, which is huge. I think this is an opportunity to test things in more clinical settings and to see what kind of how mature are some of the technology. We have a mix of things, and it is really an excellent opportunity to do so. Okay, I think it's 1,000, 5,000, or 10,000 individuals, so it's.
I think one of the main concerns when we are talking about screening tools is the easy to use for end users that they are going to be citizens. For example, in the screening of colorectal cancer, we have the physics test, and it's easy to use for end users, for citizens. They go with this kit in their house, they take, they do the test, and then they took it to the clinical setting, and it's easy for them. They don't have to have an appointment, they don't have to go to a clinical center. When we talk in breast cancer, for example, sometimes one gap is that there are concerns with harms of the test that is related to with colonoscopy happens the same. For example, the acceptance of colonoscopy for citizens is very lower when we compare with the physics test. So I think the main concern is just to try to develop easy-to-use tools for citizens and for healthcare professionals. And another main concern is the acceptance of these, that is not going to make a harm to the end users in order just to take part in those screening programs.
Thank you. So maybe we finish here not to go too late for lunch. So thank you for your attention, and I give the word to, I think, Ivan. Now, yeah. So.
So just a few words to thank you all for your excellent presentations. And I would like to thank also again to the organization because I think it was excellent. And as a key message, I would like to say just something is that we have seen people that are experts on the respective fields, multidisciplinary fields. And I think that for the success of LUCIA and for these kind of approaches, I think it's very important to speak. And it's one of the things that are probably most relevant of this project. Everyone here who have spoken and different partners in the consortium have different expertise, and we can see that it will be mutually beneficial to speak a lot, to get to the right solutions. thanks a lot, and we have the lunch here just at where we have the breakfast. Thank you very much.